Welcome to Sleepy Stories. In this episode, we will take a clear and steady look at longevity, the science of how life endures across time. The goal is to help you learn while you rest. If you do not fall asleep right away, that is completely fine. In the time you are awake, you will learn interesting scientific facts before you fall asleep. Simply listen, let your breathing slow, and allow the information to settle at its own pace. For as long as people have watched the seasons turn and counted years on their hands, there has been a quiet wish to remain alive and capable for a little longer. Legends promised fountains or potions. Philosophers searched for meaning in endurance. Modern biology offers something simpler and more reliable. Long life is not a spell. It is a disciplined management of order in a world that drifts toward disorder. Every organism faces the same challenge. Molecules scatter, structures fray, energy spreads. To live is to repair, to clean, to replace, and to coordinate those actions so that function is preserved for another day. Inside your body, this coordination happens at a scale too small to see. Each cell contains a physical record of survival strategies invented long ago. DNA stores instructions for building and maintaining the parts of life. Proteins fold into exact shapes that perform tasks from moving ions to carrying oxygen. Mitochondria transform nutrients into usable energy that powers every action. These components do not simply exist. They are continually inspected, corrected, and rebuilt. When temperature rises or a toxin slips in, protective systems respond. Damaged proteins are refolded or recycled. Broken DNA is cut out and rejoined with matching pieces. Worn cell parts are sealed inside small packages and digested, allowing the raw materials to be used again. This is maintenance, and it does not stop. Aging begins when the scale of damage grows faster than the speed of repair. That is the central equation of longevity. If repair keeps pace, function continues. If repair slows, small errors accumulate into visible decline. Science explores every step in that balance. Some lines of research examine DNA protection and the telomere caps at chromosome ends that shorten with each cell division. Other lines follow autophagy, the internal recycling system that clears debris. Still others look at inflammation, the immune response that protects against threats, but, when constant, erodes tissues over years. Across these topics the same theme appears. Longevity favors efficiency. The body that avoids waste, directs energy carefully, and rests at the right moments maintains order for longer. The environment also participates. Light and dark set daily rhythms. Temperature shifts teach the body to adapt. Food supplies wax and wane, and cells listen to those signals. When energy is abundant, growth pathways activate. When energy is scarce, repair pathways take the lead. Movement acts as a message. Muscles contract and release chemical signals that improve the health of distant organs. Sleep coordinates all of it by aligning hormones, repairing DNA, and clearing waste from the brain. None of these processes is exotic. They are ordinary and repeatable. Their power comes from their timing and their consistency. Longevity is therefore not a single secret hidden in a rare plant or a remote laboratory. It is a network of choices and responses, some made by the organism during the day, some made automatically by cells in the night. It includes what and when we eat, how often we move, the quality of the air we breathe, the steadiness of our relationships, and the way we handle stress. It also includes features we inherit at birth, such as genes that set the pace of repair and the sensitivity of our tissues to wear. Biology does not promise immortality, but it does offer leverage. Small improvements in maintenance, repeated thousands of times, become years of preserved function. In this story, we will move from origins to mechanisms to meaning. We will look at how early life learned to endure in harsh conditions and how those lessons still operate in human cells. We will examine DNA repair, protein stability, mitochondrial efficiency, and the budgeting of energy. We will consider the benefits of movement, nutrition, and sleep, and we will discuss modern attempts to influence aging with drugs, genetic engineering, and tissue replacement. We will also acknowledge limits set by physics and by the needs of societies, and we will end by asking what a longer life is for. As you listen, your own biology continues its quiet work. Enzymes patrol your DNA for mistakes. Proteins are checked for shape and function. Immune cells remove debris and send signals that calm inflammation when threats have passed. Breathing delivers oxygen that mitochondria turn into the energy needed to keep these routines going. This is the real basis of longevity, not in dramatic gestures, but in a constant, careful maintenance of order. It is reliable, it is measurable, it is already happening. The story of longevity begins long before humans existed, before even the first animals or plants. It began in an ocean filled with heat, radiation, and chemical noise. Early Earth was a dangerous place. Volcanic gases thickened the air, meteorites struck the surface, and ultraviolet light poured down without the protection of an ozone layer. Yet in that instability, life took hold. Somewhere within warm pools or deep-sea vents, clusters of molecules formed that could copy themselves. These were the earliest experiments in endurance. Some broke apart almost instantly. Others, by chance, built protective layers that kept them intact long enough to try again. At first, those molecules were fragile, held together by weak forces. To survive the chaos around them, they had to find stability. Lipids, simple fats, were among the first solutions. They spontaneously formed bubbles in water, enclosing whatever lay inside. When a useful molecule became trapped within, that bubble became a crude cell. Over time, the ones that could resist temperature shifts or ultraviolet light lasted longer. Pigments such as carotenoids and small sulfur compounds began to absorb radiation before it could destroy vital chemistry. Every successful defense increased the chance of survival, and each success was copied into the next generation. As conditions on Earth changed, so did the strategies for persistence. Molecules that could repair small breaks in their structure gained an advantage. Primitive enzymes evolved from chains of amino acids that happened to fold into shapes capable of catalyzing chemical reactions. They stitched torn fragments together, removed damaged pieces, or replaced unstable parts with stronger ones. This ability to repair rather than simply endure marked the true birth of biological longevity. Even today, the same logic governs life. To continue existing, organisms must detect and correct mistakes faster than the environment can create them. DNA, the molecule we now recognize as the blueprint of life, was a later refinement of this repair culture. Its double-stranded shape provided a backup copy. If one strand broke, the other could guide reconstruction. This redundancy transformed life's relationship with time. It meant that information could survive damage, and that memory, molecular memory, could accumulate across generations. With memory came adaptation. Organisms that remembered how to handle stress passed those lessons forward through chemistry instead of chance. Longevity became a learned behavior encoded in molecular form. The first bacteria and archaea that thrived near volcanic vents still hold traces of this ancient wisdom. Many can withstand radiation levels that would destroy most complex animals. They do so by using networks of repair enzymes that activate within seconds of exposure, sealing breaks in DNA and restoring balance. These are not modern inventions. They are the continuation of strategies billions of years old. In their presence, we see the lineage of endurance, stretching from those first chemical bubbles to every cell now alive. As multicellular life appeared, durability gained new meanings. Individual cells could die while the organism survived. Specialization emerged. Some cells focused on protection, others on reproduction, others on repair. This division of labor created resilience. Damage no longer meant extinction. It could be localized and replaced. Evolution began to favor systems capable of coordinated maintenance. Skeletons grew to provide stability. Circulatory systems evolved to deliver nutrients to distant cells. Nervous systems formed to synchronize all of it in real time. Each innovation extended the useful span of life by improving communication and resource allocation. Even the human body, despite its complexity, still echoes these primitive rules. Our cells use lipid membranes to separate inside from outside, just as the first bubbles did. Our DNA repair enzymes resemble those in simple bacteria. Our pigments, such as melanin in the skin, serve the same protective function as ancient carotenoids. We are a modern reconstruction of ancient durability, refined but not replaced. The resilience that keeps us alive today is a direct descendant of the chemistry that first organized itself against destruction. Understanding these beginnings helps explain why longevity is not a mysterious add-on but the foundation of biology itself. Life did not start strong and later weaken. It started weak and learned how to maintain. That learning never stopped. Every organism alive is the product of billions of successful repairs. The fact that you can read, think, and breathe is evidence that this unbroken chain of maintenance still holds. The same principles that governed hot oceans now govern the cells in your body. Preserve what is precious, repair what breaks, adapt to what cannot be avoided, and repeat without pause. From these simple rules, complex lives endure. That is the origin of life's durability, and it remains the essence of longevity today. Every living cell relies on a molecule that can remember, compare, and repair. DNA is often called a code, but it is more useful to think of it as stored instruction with built-in verification. Its double-stranded structure provides two matching records of the same information. When one strand is damaged, the other can guide restoration. This physical redundancy is the reason biological memory can survive across years, across generations, and across environmental shocks. The double helix is held together by base pairs with predictable affinities. Adenine pairs with thymine, cytosine pairs with guanine. During copying, specialized enzymes unzip the helix and bring in fresh nucleotides to match each exposed base. The machinery is fast, but it is not perfect. Errors occur, a wrong base is inserted. A bond breaks under oxidative stress. A strand is nicked by radiation or by normal metabolism. If no further action followed, errors would accumulate rapidly and function would collapse. Biology's answer is layered quality control. First come proofreading functions embedded in the polymerases themselves. As they add new bases, they check their own work and remove mismatches almost immediately. Next, arrive mismatch repair teams that scan the newly made DNA for subtle pairing errors and correct them before the cell proceeds. When damage is more serious, like bulky lesions caused by ultraviolet light, nucleotide excision repair cuts out the injured section and replaces it using the intact strand as a template. If both strands break, which is particularly dangerous, cells deploy double-strand break repair through non-homologous end joining or the more accurate homologous recombination, borrowing sequence information from a sister copy. Each pathway has trade-offs. Fast fixes are sometimes messy. Precise fixes take more time and coordination. Longevity depends on matching the right repair to the right injury, with minimal delay. At the ends of chromosomes lie telomeres, repetitive sequences that protect coding regions from erosion during replication. Every time a cell divides, these buffers shorten slightly. When they reach a critical length, the cell enters senescence, a protective state that halts division to prevent genomic instability. Senescent cells remain metabolically active and can secrete inflammatory factors that influence neighboring cells. A small amount of senescence is useful for tumor suppression and wound healing. Too much becomes a pro-aging signal. The body negotiates this balance with telomerase, an enzyme that extends telomeres in stem cells and certain immune cells, preserving renewal where it is most needed. In most tissues, telomerase stays quiet, trading indefinite division for genomic safety. DNA does not work alone. It wraps around histone proteins to form chromatin, which can tighten or loosen to control access to genes. Chemical tags such as methyl groups and acetyl groups mark both DNA and histones, opening some regions for active transcription and closing others for rest. This epigenetic system acts as an adjustable index to the library of DNA, telling the cell which instructions to read under which conditions. Over time, this indexing system drifts. Regions that should be silent can become noisy. Regions that should be active can become quiet. This loss of epigenetic fidelity is one of aging's signatures. Restoring proper marks, even partially, can rejuvenate cellular behavior without changing the underlying sequence. Repair and regulation require energy and coordination. Mitochondria supply ATP for the enzymes that scan, cut, copy, and rejoin DNA. When mitochondrial function falters, repair slows and errors rise. Conversely, when cells sense low nutrient availability or brief, controlled stress, they often shift toward maintenance mode, increasing autophagy and activating sirtuins that stabilize chromatin and support DNA repair. The genome and the metabolic state are therefore linked in a practical loop. Energy guides maintenance, and maintenance protects the machinery that manages energy. Not all errors should be removed. Variation is the fuel of adaptation. A small, controlled rate of mutation allows populations to adjust to changing environments. The art of longevity is to reduce harmful damage while preserving the capacity to learn. In the immune system, for example, certain cells deliberately edit their DNA to generate a diverse set of antibodies. This targeted risk strengthens defense without jeopardizing the whole. Elsewhere, strict accuracy is vital. Neurons and heart muscle cells, which may need to function for a lifetime, depend on especially careful maintenance to preserve their irreplaceable structure. When damage overwhelms repair, cells make difficult choices. They may trigger apoptosis, a tidy self-destruction that prevents malignant transformation and protects surrounding tissue. They may enter senescence to buy time for the organism, at the cost of local dysfunction. Or, if repair pathways are broken and checkpoints fail, they may slide into uncontrolled growth. Longevity research pays attention to these forks in the road because shifting decisions back toward orderly repair and away from chronic senescence or malignancy preserves function across years. DNA's endurance has a social dimension at the cellular level. In developing tissues and in adult stem cell niches, neighboring cells monitor one another through signaling molecules and adhesion contacts. A cell with damaged DNA can receive outside help, including growth factors that encourage recovery or immune signals that mark it for removal. This community approach to quality control spreads the burden of maintenance across the tissue, improving the odds that the organism stays healthy. Taken together, the picture is clear. Long life emerges when DNA is kept legible, when repair arrives on time, and when epigenetic indexing stays organized. The tools are known: polymerases that proofread, enzymes that excise lesions, pathways that mend breaks, telomeres that buy divisions, chromatin marks that coordinate expression, mitochondria that fund the work. None of these make organisms immortal. They do something more realistic and more valuable. They slow the slide from order to disorder, day after day, division after division, so that the living structure remains coherent long enough for growth, learning, contribution, and connection. This is the work happening inside you as you move through ordinary days. It is quiet and tireless. It is also the foundation for every other topic in this story, because all longevity strategies, nutrition, movement, sleep, stress management, and modern interventions, succeed only insofar as they protect and support the integrity of DNA and its repair. Inside the body, every cell behaves like a small, self-governing workshop. Its first rule is order, and its second is renewal. Mitochondria turn nutrients into usable energy, yet in doing so they also release reactive oxygen molecules that can harm nearby structures. The cell must constantly balance productivity with protection. Enzymes patrol membranes and organelles, detecting chemical wear before it spreads. When injury appears, specialized systems move in to clean and rebuild. The most important of these is autophagy, a process in which damaged components are wrapped in tiny membranes, digested, and recycled. The literal meaning is self-eating, but its purpose is preservation. Through this continual housekeeping, the cell stays young enough to function even in the midst of stress. Autophagy works in partnership with the lysosomes, acid-filled compartments that break materials down to reusable parts. Proteins that have lost their correct shapes, worn-out mitochondria, or fragments of old membranes are reduced to raw ingredients that can be built into new machinery. This constant renewal keeps the cell's internal economy efficient. Waste is converted back into resources instead of accumulating as clutter, When autophagy slows with age, debris builds up. Misfolded proteins begin to clump, communication between organelles falters, and energy becomes scarce. Cells lose flexibility, and tissues stiffen. Many degenerative diseases trace their origin to this decline in maintenance rather than to outside assault. Another system watches for larger structural failures. When damage cannot be repaired, the cell performs apoptosis, a controlled self-destruction that dismantles it safely. DNA is fragmented, membranes are sealed, and neighboring cells absorb the remnants. Though it sounds severe, apoptosis protects the organism by removing cells that might otherwise malfunction or turn cancerous. From the perspective of longevity, this sacrifice is part of the same principle, preserving the greater order even if individual components must end. It is the biological version of pruning, allowing healthy growth by removing what no longer serves. Energy management underlies all these decisions. Each act of repair requires fuel, and fuel comes from metabolism. When energy supply falls, cells shift priorities, halting division, increasing recycling, and reducing unnecessary synthesis. This metabolic slowdown is a defensive posture that can extend life at the cellular level. In animals, similar signals appear during fasting or low-calorie periods, showing that the logic of restraint operates from molecules to entire organisms. Spending less energy on growth frees resources for repair. Communication keeps the repair network synchronized. Within a tissue, cells share distress signals through molecules like ATP, calcium, and cytokines. Support cells respond by delivering nutrients or removing waste. This cooperation explains why wounds close and why organs can recover from partial injury. It also means that local maintenance reflects global conditions. Chronic inflammation or high-stress hormones can suppress autophagy and interrupt apoptosis, flooding the system with conflicting instructions. Consistency and rhythm, regular feeding, movement, and rest creates a stable environment in which maintenance processes run on time. Microscopic housekeeping extends outward to macroscopic health. When millions of cells clean and recycle efficiently, whole organs maintain elasticity and responsiveness. The liver clears toxins, the skin renews its surface, and muscles replace damaged fibers. When this cellular rhythm breaks, the effects ripple outward: fatigue, slowed healing, and vulnerability to disease. Supporting longevity, therefore, means supporting these invisible workers through adequate nutrients, balanced energy, and time for recovery. Seen at scale, the body is not a static structure but a living flow of material. Proteins you carry today did not exist months ago. Membranes are rebuilt every few weeks. Bones quietly exchange minerals. Life persists not by resisting change but by managing it so that identity is preserved while matter circulates. That is the paradox of durability, stability created through continual motion. Understanding this principle clarifies why rest and rhythm are as essential to endurance as activity and energy. Maintenance, not immortality, is the true language of longevity. All living systems run on borrowed energy. The sunlight that drives photosynthesis, the chemical bonds in food, even the oxygen in every breath, all of it must be converted, managed, and eventually paid for. Nothing in biology is free. Every act of movement, thought, or repair leaves a small trail of molecular damage behind. Understanding longevity, therefore, begins with understanding the economics of metabolism, how life spends and how carefully it accounts for the bill. Inside each cell, mitochondria convert nutrients into adenosine triphosphate, the molecule that powers almost every reaction. This process is efficient but not perfect. A fraction of electrons escape during energy transfer and react with oxygen to form reactive oxygen species, molecules so unstable that they tear through proteins, lipids, and DNA. In youth, antioxidant enzymes such as superoxide dismutase and catalase neutralize most of this chaos. With age, defenses falter and oxidative debt accumulates. The result is gradual wear that blurs the fine architecture of tissues. In this view, aging is not a mystery but compound interest on energy use. Different species manage this balance in very different ways. Small animals like mice and shrews run their metabolic engines at extraordinary speeds. Their hearts beat hundreds of times a minute. Their cells consume oxygen at rates that would exhaust a human in seconds. They live fast and die soon because the byproducts of their intensity overwhelm repair systems. Larger animals burn fuel more slowly. Elephants, whales, and tortoises stretch the same biochemical reactions across decades. Fewer free radicals per minute mean less cumulative damage. Longevity correlates loosely with metabolic rate, but not perfectly. Bats, for instance, live far longer than their size predicts. Their secret is efficiency. Mitochondria that leak fewer electrons, robust repair enzymes, and strong antioxidant signaling. Birds achieve similar feats through streamlined energy transfer and constant moderate activity rather than bursts of exhaustion. In humans, the same principles apply at subtler scales. When energy intake chronically exceeds need, mitochondria operate in surplus mode, flooding the cell with reactive byproducts and prompting inflammation. Over time, this metabolic overflow contributes to insulin resistance, cardiovascular strain, and accelerated aging. When energy is scarce, the opposite occurs. The body shifts from growth toward maintenance. Cellular sensors such as AMP-activated protein kinase and sirtuins detect low fuel and trigger autophagy, DNA repair, and antioxidant defense. The energy ledger is rebalanced in favor of preservation. Caloric moderation does not simply reduce weight, it redirects biochemical priorities toward survival. The link between energy and lifespan extends to behavior. Periodic movement enhances efficiency by stimulating mitochondrial turnover, ensuring that old, leaky organelles are replaced with new ones. Regular sleep consolidates energy budgets across tissues, synchronizing hormone release and glucose use. Even psychological calm conserves resources. Chronic stress keeps metabolism in fight or flight mode, draining reserves meant for repair. In this sense, longevity represents not stillness but rhythm, alternating expenditure with recovery, so that no system runs past its limits for long. Nature reinforces this rule through feedback. If an organism spends too freely, damage signals rise, activating inflammatory and apoptotic pathways that curtail function. If it spends too little, essential renewal halts, and decline follows from neglect. The long-lived strike a narrow balance, active enough to keep systems responsive, restrained enough to avoid waste. Whales migrate thousands of kilometers but glide rather than sprint. Trees grow slowly yet persist for centuries by distributing energy evenly among roots, leaves, and protective bark. Longevity favors those who move with precision rather than intensity. Human cultures mirror these biological patterns. Societies with traditional eating rhythms, moderate portions, periods of fasting, reliance on unprocessed foods, tend to exhibit lower rates of metabolic disease and longer average life. The pattern suggests that restraint woven into daily routine becomes physiology over time. Energy wisdom is learned both by bodies and by cultures. At its deepest level, metabolism is a conversation between order and entropy. Every joule extracted from food must be dissipated as heat or stored temporarily in chemical bonds. No system escapes that law. What life achieves, remarkably, is to borrow energy just long enough to maintain structure before returning it to the environment. The art of longevity is to keep this borrowing sustainable, to stretch the cycle so that structure persists without collapse. The fewer unnecessary reactions a body performs, the less collateral damage it must repair, and the more time it can allocate to renewal. In this way, energy becomes the silent measure of time. A heartbeat, a breath, a thought, all are tiny expenditures from a finite account. Yet within that limit lies freedom. Through balance and rhythm, the same energy that could burn life quickly can instead sustain it gracefully. Longevity is the discipline of that conversion, the decision, repeated in every cell, to spend wisely rather than wastefully, to power life just enough to keep its order intact. Within every cell, proteins perform nearly all the work that keeps life running. They form the scaffolding of tissues, transport nutrients, carry messages, and drive the chemical reactions that sustain metabolism. To accomplish so many tasks, each protein must fold into a precise three-dimensional shape. The folding process happens spontaneously, guided by electrical attractions between atoms. When everything goes well, the result is a structure that fits perfectly into its biological role. When the process misfires, the consequences ripple outward through the organism. Protein misfolding is a constant threat because the shapes are fragile. Heat, radiation, toxins, and even normal oxidative stress can twist a protein out of form. Once distorted, it may clump with others, forming aggregates that interfere with cellular machinery. The body has evolved an elaborate network of guardians to prevent this. Among the most important are the chaperone proteins, sometimes called heat shock proteins, which act as molecular attendants. They bind to newly made or damaged proteins, helping them refold into proper shapes or escorting them to disposal if repair fails. When temperature rises or stress increases, cells produce extra chaperones, anticipating the wave of unfolding to come. This adaptive surge demonstrates the cell's priority: protect structure first, handle growth later. Proteins that cannot be saved are targeted for degradation. A system called the ubiquitin proteasome pathway labels unwanted molecules with a small tag of ubiquitin. The tagged proteins are fed into a cylindrical complex, the proteasome, where they are chopped into short peptides and recycled. This recycling recovers amino acids for new synthesis and prevents buildup of defective material. When the system slows with age, damaged proteins accumulate faster than they can be cleared. The resulting aggregates, such as beta-amyloid plaques or tau tangles, disrupt neurons and are hallmarks of degenerative diseases. Similar clumps appear in muscles and liver cells, reducing elasticity and function. These are not isolated failures, but signs that molecular housekeeping is losing pace. To counteract wear, cells rely on both precision and flexibility. Precision ensures that vital enzymes keep their shapes. Flexibility allows temporary distortions to be corrected. Many proteins are intrinsically disordered, shifting form depending on context. Their fluidity allows quick adaptation to changes in temperature, pH, or salt concentration. This balance between structure and movement is crucial for resilience. Too rigid, and proteins crack under stress. Too loose, and they cannot perform specific tasks. Long-lived organisms often express variants of proteins with extra stabilizing bonds or more efficient chaperone systems, reinforcing the correlation between molecular sturdiness and lifespan. Energy fuels these corrections. Folding, refolding, and degradation all require ATP, supplied by mitochondria. When energy runs low, maintenance falters. Cells faced with energy shortage triage their efforts, focusing on the most critical repairs. If shortages persist, misfolded proteins overwhelm the system, activating a stress response in the endoplasmic reticulum known as the unfolded protein response. This signaling cascade halts protein synthesis, boosts chaperone production, and, if stress remains unresolved, initiates cell death. The choice between restoration and self-destruction again illustrates the core logic of longevity: stability preserved through timely, decisive intervention. External factors influence internal order. Diets rich in antioxidants and trace minerals supply raw materials for protective enzymes, while persistent toxins such as heavy metals disrupt folding chemistry. Heat and exercise, paradoxically, strengthen molecular defenses by provoking mild stress that upregulates chaperones and proteasomes. The pattern mirrors other hormetic effects observed throughout biology: short, controlled challenges that reinforce durability rather than erode it. Viewed at the scale of an organism, protein maintenance underpins visible vitality. Collagen keeps skin and tendons supple. Enzymes maintain digestion. Hemoglobin delivers oxygen efficiently. Antibodies guard against infection. When these molecules remain intact and responsive, tissues act younger than their chronological age. When their architecture fails, aging becomes apparent in stiffness, fatigue, and cognitive decline. Molecular stability thus links directly to physical resilience. Life's persistence depends not on avoiding stress but on handling it gracefully. Proteins are the front line of that negotiation, constantly folding, unfolding, and refolding in a rhythm as steady as breathing. Each correction buys time. Each successful clearance renews order. The lesson is consistent from molecule to organism. Longevity is achieved not by perfection but by an unending willingness to repair. Beneath every act of renewal lies this quiet chemistry of folding and patience, a choreography of order against entropy that defines what it means to stay alive. Every organism faces change. Temperature rises, food supply shifts, predators appear, or oxygen levels fall. To survive these fluctuations, life evolves systems that transform stress into strength. This principle, called hormesis, describes how mild challenges trigger adaptive responses that leave the organism more resilient than before. The same heat, fasting, or exercise that would destroy a weakened cell can, in the right dose, awaken dormant repair pathways. Longevity thrives on that balance. Enough stress to train, not so much to break. At the cellular level, hormesis begins with sensing. Specialized proteins detect shifts in temperature, oxidation, and nutrient status, then signal protective genes to activate. A brief rise in reactive oxygen molecules, for example, stimulates antioxidant defenses. Temporary shortage of glucose activates AMP-activated protein kinase, shifting metabolism toward energy conservation and repair. Mechanical strain on muscles releases growth factors that strengthen connective tissue and bones. Even psychological challenges, when contained and recoverable, sharpen focus through transient surges in adrenaline and dopamine. These micro-stresses expand the range of conditions under which the body can function comfortably. Evolution has refined this calibration. Early life experienced constant physical extremes: heat vents, freezing tides, radiation, and scarcity. Species that could mount flexible stress responses survived. The descendants of those survivors, including humans, carry molecular memories of those trials. Inside every cell, stress response elements lie dormant until provoked. Once activated, they drive production of heat shock proteins, DNA repair enzymes, and antioxidants. When the challenge passes, feedback loops shut them down again, preventing waste and maintaining equilibrium. This cycle of activation and rest is the rhythm of endurance, a biological form of training that keeps maintenance systems alert. The same principle applies to higher organisms. Exercise exemplifies hormesis in motion. Muscle contractions create oxidative stress and small tears in fibers, prompting repair and growth that leave the tissue stronger. Cardiovascular strain increases blood flow and stimulates mitochondrial biogenesis, improving efficiency of oxygen use. Overtraining, however, flips the balance, leading to chronic inflammation and exhaustion. The line between strengthening and damaging is thin, reminding that longevity depends as much on recovery as on challenge. The most durable species and individuals practice this rhythm naturally, alternating stress with rest. Fasting works through similar dynamics. When nutrients are scarce, cells shift from reproduction to preservation, turning on autophagy and other cleanup pathways. After the fast ends, nutrient-sensing pathways such as mTOR reactivate, rebuilding the cleaned infrastructure. Cycles of scarcity and abundance maintain metabolic flexibility, the ability to switch smoothly between energy sources without distress. Modern constant abundance, by contrast, keeps growth signals permanently on and repair signals quiet, hastening metabolic fatigue. Strategic pauses in consumption restore the natural rhythm that evolution assumed. Thermal stress, too, has its uses. Cold exposure increases circulation and encourages brown fat activity, improving energy efficiency. Heat exposure from saunas or hot climates induces protective proteins and improves vascular health. The benefits arise not from comfort but from controlled discomfort, brief enough to activate defenses without inflicting damage. Across cultures, traditional practices, bathing, fasting, physical labor, echo these adaptive lessons learned long before science named them. On the molecular scale, hormesis trains the repair networks described earlier. Each mild challenge rehearses the cell's response to larger threats, keeping enzymes, chaperones, and immune messengers ready. Without occasional rehearsal, these systems grow complacent, much like muscles unused for too long. Laboratory studies confirm this. Organisms kept in sterile, unchanging environments often lose resilience, while those exposed to variable conditions adapt more vigorously and live longer. The psychology of adaptation mirrors the biology. Humans who face manageable difficulties and recover grow more confident and emotionally stable. Chronic, uncontrollable stress, however, erodes both mind and body, keeping hormones like cortisol elevated and suppressing immunity. The distinction again lies in scale and recovery time. Whether in cells or consciousness, health depends on challenge followed by calm restoration. Ultimately, Hormesis explains why longevity does not arise from avoidance of hardship, but from measured engagement with it. Life improves through correction, not insulation. Muscles, mitochondria, neurons, and even thought patterns follow the same rule. Without stimulus, they decline. With appropriate challenge, they strengthen. The art of living long is to master this rhythm, to invite stress deliberately, absorb its lesson, and then return to rest. In that oscillation between effort and recovery, organisms find the enduring equilibrium that evolution has written into every cell. Nutrition is the conversation between the body and its environment. Every bite delivers information, not just energy. Within that information are the molecules that tell cells whether to grow, repair, or rest. Food is chemistry shaped by evolution. Metabolism is the interpretation of that chemistry. When these two remain in dialogue, the body stays in balance. When the conversation becomes noisy, too much sugar, too few micronutrients, irregular timing, cellular communication falters, and aging accelerates. Modern biology now describes how nutrients control the master switches of longevity. The mTOR pathway senses amino acids and triggers growth and protein synthesis. The AMP-activated protein kinase AMPK pathway monitors low energy and signals cells to recycle and conserve. The sirtuin family of enzymes responds to the ratio of energy carriers NADMIs, NADH, coordinating DNA repair and mitochondrial efficiency. Together, these networks decide whether the body invests in expansion or in maintenance. When energy and protein are abundant, mTOR dominates, favoring growth and reproduction. When calories decline, AMPK and sirtuins take the lead, focusing on repair and preservation. Longevity flourishes when this seesaw tilts periodically toward maintenance rather than being locked in constant growth. The macronutrients, carbohydrates, fats, and proteins each carry their own signaling weight. Glucose provides quick fuel but spikes insulin, the hormone that directs cells to store energy. Persistent high insulin keeps growth signals on and repair genes off, shortening health span. Dietary fats supply dense energy and serve as raw material for membranes and hormones. The balance between saturated and unsaturated fats influences inflammation and cellular fluidity. Proteins supply amino acids, but excess intake, particularly of branched-chain amino acids, overstimulates mTOR. Moderation and diversity therefore matter more than exclusion. A diet slightly lower in protein but rich in varied plant sources often supports the longevity pathways that prioritize repair. Micronutrients, vitamins, minerals, and phytochemicals act as catalysts. Zinc and magnesium stabilize DNA repair enzymes. Vitamin C and E neutralize free radicals, while polyphenols from fruits, vegetables, tea, and olive oil activate protective genes through mild stress signals. Resveratrol, curcumin, and catechins are famous examples. They imitate scarcity at the molecular level, nudging cells toward maintenance without true deprivation. The lesson is elegant. Certain plant compounds evolved as defense chemicals, and when humans consume them, those same molecules provoke adaptive strength. Timing adds another dimension. The human body follows circadian rhythms that govern hormone release, digestion, and cell division. Eating at consistent times aligns metabolism with these internal clocks. Late-night meals, when the body expects rest, disrupt insulin cycles and elevate blood sugar through the night. In contrast, daylight eating followed by an overnight fasting period improves insulin sensitivity and lipid balance. This time-restricted feeding does not reduce total calories but synchronizes intake with metabolism's natural schedule, allowing nightly maintenance to proceed undisturbed. Hydration, often overlooked, supports every biochemical exchange. Water regulates temperature, transports nutrients, and removes waste through kidneys and lymph. Even mild dehydration slows enzymatic reactions and impairs energy production. Long-lived populations tend to consume abundant plain water or mineral-rich herbal infusions, maintaining circulation without excess calories. Cultural patterns echo biological truths. Traditional diets in regions of exceptional longevity, Mediterranean, Okinawan, Nicoyan, share core features, mostly plant-based foods, modest portions, fermented staples that nurture gut microbes, and minimal refined sugar. These diets naturally modulate the same molecular pathways scientists now identify in laboratories. They also embed eating in social rhythm, shared meals, unhurried pace, gratitude before consumption. Emotional context matters. Digestion begins in the mind, where calm activates the parasympathetic nervous system that coordinates nutrient absorption and repair. Malnutrition at either extreme undermines endurance. Chronic scarcity weakens immunity and slows repair, while chronic excess clogs the same pathways with constant signaling noise. Balance, the recurring theme of longevity, applies here too. Variety supplies all building blocks, moderation prevents overload, and periodic light scarcity invites renewal. What we eat shapes not only our tissues but the tempo of our metabolism, deciding how quickly or slowly we trade energy for time. In essence, nutrition teaches the same lesson found throughout biology. Cycles sustain life. Alternation between abundance and restraint, feeding and fasting, activity and rest keeps the machinery tuned. The molecules of food are messages from the outside world to the inner one, and reading those messages accurately determines how long the conversation continues. Longevity listens carefully, choosing foods that whisper repair rather than shout growth. Longevity wears many faces in nature. Some creatures live for hours, others for centuries, yet all obey the same physical laws. Comparing them reveals which biological designs stretch time and which spend it quickly. Evolution, by experimenting through countless generations, has already tested most of the possibilities for endurance. Each species carries lessons about how structure, metabolism, and environment combine to define the tempo of life. Among the most enduring animals on Earth are the bowhead whales, inhabitants of the Arctic seas. Genetic and chemical analyses of their tissues show ages exceeding 2 centuries. Their secret lies in slow metabolism, cold environments, and remarkably efficient DNA repair. Mutations in their cellular stress response genes enhance maintenance over growth, and their low body temperature reduces oxidative damage. Despite their size, or perhaps because of it, they move through life at a deliberate pace, spending energy sparingly in an environment that demands patience. On land, Galapagos, tortoises, and Aldabra giants live more than 100 years, supported by a sluggish metabolism and a lifestyle defined by rest and steady consumption. Their hearts beat a few dozen times per minute. Their cells divide infrequently. Less activity means fewer opportunities for errors to accumulate. The same principle applies to koi fish in tranquil ponds and Greenland sharks drifting through icy waters. Slow motion preserves structure. By minimizing heat and turbulence, these species stretch biological time like a slow heartbeat extending through centuries. At the microscopic scale, other organisms take the opposite approach. Some bacteria can suspend activity entirely, forming spores that endure drought, heat, or radiation for thousands of years. When conditions improve, they awaken as if no time had passed. Tardigrades, the tiny water bears, enter a similar cryptobiotic state by replacing water in their cells with protective sugars that prevent crystallization. These forms of suspended animation demonstrate that survival need not mean continuous activity. Sometimes longevity is achieved through strategic stillness. Perhaps the most fascinating model of natural immortality is the hydra, a small freshwater polyp that continually renews its cells through perpetual stem cell activity. Under stable conditions, hydra show no measurable aging, their risk of death does not increase with time. They appear to sidestep senescence altogether. A related story unfolds in certain species of planarian flatworms, which can regenerate an entire body from a single piece. Their stem cells, called neoblasts, remain pluripotent throughout life, restoring tissues indefinitely. These examples suggest that biological aging is not inevitable but context-dependent, an outcome of energy allocation, repair efficiency, and genetic regulation. Even within mammals, lifespan varies dramatically. Mice live a few years, naked mole rats survive more than 30, resisting cancer and maintaining protein stability far longer than expected. Their secret includes high molecular weight, hyaluronan that protects cells from crowding damage, enhanced proteasome activity, and constant expression of chaperone proteins. Birds, though warm-blooded and metabolically intense, outlive mammals of similar size. Their mitochondria produce fewer free radicals, and their efficient flight muscles distribute oxygen evenly, reducing local oxidative stress. Human longevity sits midway among these extremes. Our maximum lifespan, around 120 years, reflects a balance between energy expenditure, repair capacity, and social complexity. We are neither the slowest nor the fastest metabolizers, but we excel at environmental control. Shelter, medicine, and cooperation reduce external hazards, allowing our internal repair systems to become the main determinant of life length. Cultural evolution extended biological endurance by stabilizing surroundings faster than genes could. Across all these examples, several patterns emerge. Long-lived species usually share low reproductive rates, stable environments, and robust maintenance mechanisms. Short-lived species trade endurance for speed, producing many offspring to offset high mortality. Evolution weighs energy budgets differently depending on risk. When external danger is high, quick reproduction wins. When safety increases, maintenance pays off. The biological strategies mirror economic ones, high turnover versus long-term investment. Studying these patterns gives scientists both inspiration and caution. Genes that extend lifespan in worms or flies rarely act the same way in humans, whose complexity changes the equation. Still, the comparative view clarifies what works. Minimize waste, manage energy slowly, prevent molecular clutter, and favor quality over quantity. Nature's centenarians, from whales to tortoises to humble hydra, share this quiet discipline. Their lesson is simple: life lengthens when movement, energy, and repair are in harmony with environment. In the stillness of deep oceans or the unhurried climb of a tortoise, we see not slowness but control, the steady rhythm that keeps order intact for as long as physics allows. Longevity, it seems, is the art of pacing, practiced differently by every species, yet governed by the same enduring laws. Inside every organism is a system of timing that decides when to grow, when to reproduce, and when to slow down. This timing is not measured in minutes or hours, but in molecular signals that flow between genes. The result is what scientists call the genetic clock, a network of pathways that governs maintenance and decline. Aging, in this view, is not random collapse but a gradual reprogramming of biological priorities. The body, following ancient evolutionary rules, invests heavily in survival until reproduction is secure, then gradually shifts resources away from repair. Longevity research seeks to understand, and where possible, rebalance that program. One of the central circuits in this clock is the insulin, IGF-1 signaling pathway. In simple organisms such as worms and flies, reducing IGF-1 activity dramatically extends lifespan. Lower nutrient signals activate protective genes, including those controlled by the FOXO family of transcription factors. These genes improve DNA repair, increase antioxidant defenses, and enhance autophagy. In mammals, the pattern persists but is more complex. High insulin and IGF-1 encourage growth during youth, while persistent activation later in life accelerates aging through chronic inflammation and metabolic stress. A moderate decrease in this signaling, achieved through diet or pharmacological agents, can mimic the beneficial effects of calorie restriction, nudging the clock towards slower ticking. Another component is the mTOR pathway, which interprets amino acid abundance as a command to build. When constantly active, mTOR keeps cells in growth mode, diverting energy from repair. Inhibiting mTOR with compounds such as rapamycin extends lifespan in multiple species by re-establishing balance. Yet suppression must be partial. Growth and immunity still require its activity. The key is modulation, not elimination, precisely the kind of fine-tuning longevity depends on. The clock also runs through the sirtuin family of enzymes, which sense the cell's energy state via NADMYs levels. When energy is low, sirtuins trigger chromatin remodeling, stabilize DNA, and improve mitochondrial function. In mammals, 7 sirtuins coordinate different aspects of repair, stress resistance, and metabolic flexibility. Their activity declines with age as NADMYs become scarce, linking energy metabolism directly to the pace of aging. Restoring NADMYs levels through lifestyle or supplementation is one of the most studied strategies in current longevity science. Beyond metabolism, genes manage repair timing through hormonal and circadian coordination. The pineal gland releases melatonin in darkness, aligning internal rhythms with day and night. Cortisol and growth hormone follow similar cycles. Disruption of these rhythms, by erratic sleep or constant artificial light, confuses the genetic clock, leading to metabolic drift and premature aging. Aligning behavior with natural light cycles allows genes and hormones to operate in synchrony, maximizing the efficiency of repair during rest. At the deepest level, this molecular timing evolved from a practical logic. An organism that invests infinite energy in maintenance would reproduce too slowly to survive as a species. The trade-off, known as the disposable soma theory, states that evolution optimizes survival just long enough for reproduction and care of offspring, not for indefinite perfection. Humans, through social cooperation and medicine, have extended the benefits of maintenance beyond that boundary, but the underlying biology still follows its ancestral schedule. Longevity science aims to rebudget the system, to redirect some energy from short-term growth back to long-term stability without disturbing essential functions. Epigenetic changes further regulate the clock. Over time, chemical marks on DNA accumulate in predictable patterns, allowing researchers to estimate biological age from them. These epigenetic clocks provide a measurable link between gene regulation and live time. Some studies show that lifestyle adjustments, diet, exercise, stress reduction can modestly reverse these markers, suggesting that the clock can be reset, at least partially. Ultimately, the genetic clock represents a conversation between inheritance and experience. Genes provide the rhythm, environment and behavior determine the tempo. While we cannot stop the clock, we can influence its speed by managing the signals it listens to: nutrient flow, circadian light, physical activity, and emotional stability. Each adjustment adds a fraction of time, not by tricking biology but by harmonizing with its natural cadence. In the steady ticking of this molecular machinery lies both limitation and opportunity. The clock reminds that life's duration is negotiated, not decreed. When its gears move in sync with our choices, time becomes elastic, stretching quietly through balance, restraint, and respect for the rhythm that built us. That rhythm is the real heartbeat of longevity. Hormones are the body's chemical timekeepers. They drift through the bloodstream like messages in a vast internal network, coordinating metabolism, mood, growth, and repair. Each molecule acts locally yet affects the entire organism, setting the pace at which energy is used and tissues are renewed. Because hormones operate in cycles, longevity depends on the stability of their rhythm as much as on their strength. When the orchestration falters, the tempo of aging quickens. The endocrine system includes glands scattered throughout the body: pituitary, thyroid, adrenals, pancreas, gonads, each releasing substances that influence distant cells. The pituitary gland, often called the master regulator, dispatches growth hormone and controls others through its signaling. In youth, growth hormone surges, driving development and repair. With age, its nightly pulses diminish, slowing tissue renewal and reducing muscle mass. Excessive decline leads to frailty, but excessive replacement carries risks of imbalance and cancer. Longevity lies in moderation. Enough signal to sustain maintenance, not so much that the system burns energy on endless expansion. The thyroid governs metabolic speed. Its hormones, thyroxine, T4, and triiodothyronine, T3, determine how quickly cells convert fuel into energy. Overactive thyroids accelerate metabolism and wear down organs. Underactive ones leave tissues sluggish and cold. Studies of long-lived populations reveal slightly lower thyroid activity, suggesting that a gentler metabolic rate conserves resources and reduces oxidative damage. This pattern aligns with nature's larger lesson, that slower rhythms often correlate with longer life. At the adrenal level, the conversation centers on stress. The glands perched above the kidneys release cortisol and adrenaline in response to pressure or danger. Short bursts sharpen focus and mobilize glucose for quick energy. Chronic elevation, however, keeps the body in alert mode, suppressing immunity and degrading collagen, leading to premature aging of skin, vessels, and organs. The relationship between stress and longevity follows a curve. Too little stimulation breeds weakness, too much erodes resilience. Recovery time, the return to calm, is the crucial variable that determines whether stress strengthens or shortens life. Insulin, produced by the pancreas, is another central hormone in the longevity equation. It ushers glucose into cells for use or storage. Persistent surges from high-sugar diets make tissues resistant to its signal, forcing ever higher production and fueling inflammation. Moderating carbohydrate intake and maintaining muscle activity restore sensitivity, ensuring that energy flow remains smooth and repair pathways stay active. The same hormone that nourishes can also age, depending on consistency and scale. Sex hormones, estrogen, progesterone, and testosterone, connect reproduction with repair. During reproductive years, they sustain bone density, muscle tone, and vascular health. Their decline in midlife marks a shift from reproduction to preservation. The body, having completed its evolutionary mandate, reduces investment in maintenance. Modern medicine can partially offset this loss, yet evidence suggests that lifestyle factors, movement, diet, and rest influence these hormones more safely than pharmacology alone. Balance achieved naturally integrates better with the body's internal rhythms. Another messenger, melatonin, bridges endocrine control with circadian timing. Secreted by the pineal gland during darkness, it cues sleep and coordinates nightly repair. Beyond its role in rest, melatonin acts as an antioxidant, stabilizing mitochondria and reducing DNA damage. Exposure to artificial light at night suppresses its release, confusing metabolic schedules. Re-establishing darkness and consistent sleep hours restores this ancient signal and indirectly supports longevity by allowing the repair machinery to work uninterrupted. The hormonal system is both delicate and adaptable. It listens constantly to cues from the environment, light, temperature, food, and social interaction, and adjusts secretion patterns accordingly. When life becomes irregular, hormones echo the chaos. When routines stabilize, they settle into rhythm. Meditation, regular exercise, and steady meal times all contribute to endocrine harmony. Such alignment is not merely psychological, it translates into measurable biochemical order: lower cortisol, steadier insulin, improved thyroid and sex hormone balance. In essence, hormones are the body's internal clock, tuned not by ticking gears but by feedback loops of chemistry. They remind that longevity is a matter of synchronization, between organs, between day and night, between effort and rest. When the signals flow in sequence, every tissue receives what it needs, and energy is used with precision. Aging then becomes not a failure of the system, but a gradual modulation of tempo. To live long is to keep that orchestra in tune, letting each note fade and return in balance with the next. The brain is both the architect and the mirror of aging. It directs the body's rhythms through hormones and nerves, yet it also ages in response to the body's changing chemistry. Unlike most tissues, neurons rarely divide. They must last a lifetime. Longevity, therefore, depends on how well the brain protects, repairs, and reorganizes itself over time. Aging here is not only loss, it is the gradual reshaping of neural networks to preserve coherence as the physical world erodes around them. Neurons communicate through electrical pulses and chemical messengers called neurotransmitters. Each connection, or synapse, forms a bridge of memory and learning. Over decades, these bridges experience wear. Oxidative stress, inflammation, and reduced blood flow damage their delicate membranes. Mitochondria within neurons weaken, producing less energy and more free radicals. The result is slower signaling, lapses in recall, and reduced coordination. Yet the brain compensates through neuroplasticity, its ability to rewire circuits, recruit alternate pathways, and strengthen surviving connections through practice and novelty. Mental challenge and engagement act as biological training, stimulating growth factors such as brain-derived neurotrophic factor, BDNF, that repair and reinforce synapses. Blood supply is critical to this maintenance. The blood-brain barrier filters what enters neural tissue, but with age it becomes more permeable, allowing inflammatory molecules inside. Regular physical activity counteracts this process by improving circulation and oxygen delivery. Exercise increases levels of nitric oxide, relaxing blood vessels and enhancing nutrient flow to neural tissue. Studies consistently show that active individuals maintain larger hippocampal volumes, the regions responsible for memory, than sedentary peers. Movement, though rooted in muscle, is nourishment for the mind. Sleep is the brain's main maintenance window. During deep stages, neurons reduce activity while the glymphatic system flushes waste, such as beta-amyloid and tau proteins. When sleep is shortened or fragmented, this cleaning fails, and debris accumulates. Chronic deprivation accelerates cognitive decline. Aligning sleep with natural circadian rhythms, dark nights, consistent schedules, minimal artificial light, keeps this nightly restoration efficient. Memory consolidation, emotional regulation, and creativity all depend on it. Nutrition also reaches the brain indirectly. Omega-3 fatty acids from fish or algae maintain membrane flexibility, allowing signals to pass smoothly. Antioxidants from berries, leafy greens, and cocoa protect against oxidative injury. Excessive sugar, in contrast, stiffens blood vessels and increases inflammation, diminishing mental performance. The gut microbiome, once considered unrelated to the brain, now appears to influence mood and cognition through chemical messengers that travel along the vagus nerve. Feeding beneficial microbes supports not only digestion but neural health, linking the two ends of the body's internal conversation. Emotional experience further shapes neural aging. Chronic psychological stress releases cortisol, which shrinks dendritic branches in the hippocampus and disrupts memory formation. Mindfulness, social connection, and creative activity reverse these trends, promoting calm neurochemistry that favors repair. The human brain evolved to respond to challenge, followed by recovery. Modern life often provides constant challenge with little recovery. Restoring cycles of effort and relaxation allows neurotransmitters to reset and prevents the exhaustion of attention that feeds anxiety and insomnia. Electrical rhythm is another subtle aspect. Brain waves, alpha, beta, theta, delta, synchronize communication between regions. Age and distraction desynchronize these patterns, reducing efficiency. Meditation, rhythmic breathing, and music can restore coherence, aligning internal tempo with external stimuli. The result is subjective, calm, and measurable order, a physiological echo of harmony repeated throughout this exploration of longevity. At the molecular level, neurons defend themselves with repair enzymes similar to those in other cells, but their long life gives them fewer chances to start over. Protecting these cells means preserving the body's identity. Even when memory fades, the underlying personality, humor, and sense of self often persist, evidence that deep neural structures remain resilient. In sum, the aging brain teaches a paradox. It loses cells but gains wisdom in how to use what remains. Longevity does not depend on freezing time but on continual adaptation, building new roots around lost bridges, deepening connections that matter, discarding those that no longer serve. Each thought, emotion, and choice reshapes the organ that houses them. When activity, rest, nutrition, and calm interlock, the brain's internal weather clears, revealing a stable climate capable of supporting long life in both body and mind. The immune system is the body's protective intelligence. It identifies what belongs, what does not, and what must be repaired. Without it, life would last only moments. Yet the same force that defends can also destroy if left unchecked. Longevity depends on how precisely the immune system distinguishes friend from foe, and how calmly it retreats once a battle is won. This defense network has two layers: the innate immune system, fast and general, and the adaptive system, slower but highly specific. The innate response acts first, sending macrophages and neutrophils to engulf invaders and release chemical signals that summon reinforcements. The adaptive response follows, producing targeted antibodies and memory cells that remember the threat for future encounters. Together, they form a dynamic surveillance grid that spans every organ, constantly exchanging information through cytokines, molecular messengers of alarm and resolution. When young, this coordination is nearly perfect. Inflammation rises quickly to contain damage, then subsides as healing begins. With age, the shutdown becomes incomplete. Low-level inflammation lingers, producing a condition scientists call inflammaging. The immune system, designed for emergencies, starts to misfire in peacetime. Cytokines remain elevated, generating oxidative stress that erodes blood vessels, joints, and nerves. Repair slows because resources are spent maintaining an unnecessary state of alert. The body, in essence, wages a quiet civil war against itself. Several factors drive this drift. Over decades, immune cells accumulate DNA damage and metabolic fatigue. The thymus gland, where T cells mature, shrinks after adolescence, reducing the diversity of available defenders. Chronic infections, such as cytomegalovirus, keep parts of the system permanently engaged, depleting reserves. Lifestyle amplifies these effects. Poor diet, sleep loss, pollution, and psychological stress all prolong inflammatory signaling. Each contributes a whisper of irritation that, when combined, becomes a constant background hum. Nutrition modulates this process profoundly. Diets rich in omega-3 fatty acids, antioxidants, and polyphenols lower cytokine production and enhance immune resolution. Excess sugar and processed fats do the opposite, sustaining oxidative reactions that damage tissues. Fiber supports gut microbes that train immune cells to differentiate between harmless and dangerous stimuli. Since 70% of immune cells reside near the intestines, the gut is effectively a classroom where tolerance and restraint are learned daily. Movement is another regulator. Moderate exercise mobilizes white blood cells and increases circulation of anti-inflammatory molecules. Intense, prolonged exertion without recovery, however, suppresses immunity temporarily. Balance again defines the outcome. Sleep provides the final reset. During deep stages, cortisol drops and melatonin rises, promoting the release of growth hormone and immune peptides that restore equilibrium. Irregular or shortened sleep keeps inflammation simmering through elevated stress hormones. Psychology influences immunity through the brain's communication with the endocrine system. Chronic worry or social isolation increases sympathetic nerve activity, which instructs bone marrow to produce more inflammatory cells. By contrast, social connection, gratitude, and calm breathing signal safety, allowing the body to divert resources from defense to renewal. The link between emotional peace and physical resilience is now measurable in levels of cytokines and antibody response. Modern science seeks to fine-tune immunity rather than suppress it. Senolytic therapies aim to remove aged immune cells that secrete inflammatory factors, while immunomodulatory drugs attempt to restore youthful balance without blunt force. Vaccination extends adaptive memory, preventing infections that would otherwise accelerate systemic wear. None of these innovations replace the fundamentals: nutrition, movement, rest, and mental stability, but they complement them, addressing the accumulated noise that time introduces. In the context of longevity, the immune system embodies the balance between vigilance and peace. It must respond swiftly yet withdraw gracefully, defend without destroying, Remember without obsessing. Aging is not the result of battle alone, but of the inability to end the battle. When inflammation learns again to rise and fall with precision, the body recovers its natural rhythm of protection and repair. That rhythm, neither too loud nor too quiet, is the heartbeat of enduring health. Longevity is not shaped by biology alone. The body exists in a constant dialogue with its surroundings: light, air, temperature, microbes, and the countless materials we touch and breathe. Every cell reflects this conversation. The longer and cleaner that dialogue remains, the slower aging proceeds. The environment can either sharpen or dull the tools of maintenance that life depends on. Sunlight is the oldest environmental signal. It synchronizes circadian rhythms, drives vitamin D production, and regulates hormone release. Moderate exposure supports bone strength and immune balance, while overexposure causes DNA damage and oxidative stress in the skin. Melanin, sunscreen, and repair enzymes evolved as natural shields against ultraviolet radiation. The lesson is typical of longevity's logic: protection, not avoidance. A few minutes of daylight each day calibrates the body's internal clock, improving sleep and metabolic harmony. Air, the medium of respiration, carries both nourishment and threat. Clean air feeds mitochondria with oxygen. Polluted air introduces fine particles that inflame lungs and vessels. These particles trigger immune reactions similar to infection, accelerating vascular aging and cognitive decline. Studies show that people living near green spaces or coastlines, where air is cleaner and humidity steadier, have lower rates of chronic disease. Trees, by filtering air and releasing phytoncides, create microclimates that calm the nervous system and reduce stress hormones. The biology of longevity extends, quite literally, into the quality of the atmosphere we share. Water tells a similar story. Every organ depends on its purity and mineral balance. Contaminants such as heavy metals, pesticides, or microplastics accumulate silently, disrupting hormones and cellular repair. Yet the solution is not isolation from the world, but stewardship of it. Environments that sustain life sustain health. Regions with mineral-rich groundwater, moderate temperatures, and stable ecosystems consistently produce longer-lived populations. Their biology and geography cooperate instead of compete. Temperature shapes endurance through adaptation. Mild, cold exposure activates brown adipose tissue, which burns energy to maintain warmth and improves metabolic flexibility. Heat, when controlled, promotes circulation and detoxification through sweat. Extreme or chronic exposure to either end exhausts resources, but alternating warmth and coolness trains vascular resilience. Traditional practices, bathing, sauna, swimming, echo this wisdom. They expose the body to transient stress that awakens dormant maintenance pathways, similar to hormesis at the cellular level. Noise, light pollution, and artificial environments introduce subtler forms of stress. Constant noise raises cortisol. Artificial light at night disrupts melatonin, confusing the brain's sense of time. Even visual clutter and electromagnetic signals contribute to a background tension that keeps physiology slightly alert. Long-lived cultures often live closer to natural cycles. Early sunrise, communal meals at dusk, quiet evenings. Their surroundings reinforce the same rhythm biology expects. Microbial exposure is another hidden factor. Urban cleanliness, while protecting against infection, can deprive the immune system of the diversity it needs to learn tolerance. Contact with soil, animals, and plants trains immunity to react proportionally instead of excessively. The old friends hypothesis proposes that exposure to harmless microbes early in life programs calm immune behavior later on. Balanced hygiene, cleanliness without sterility, preserves this education. Climate change and environmental degradation threaten these delicate supports of longevity. Rising temperatures strain cardiovascular systems. New pathogens challenge immunity. Pollution undermines cellular integrity. Protecting personal health thus merges with protecting planetary health. The body and the biosphere are continuous systems exchanging air, water, and light. Damage to one accelerates aging in the other. When people migrate to healthier environments, less pollution, more greenery, slower pace. Biomarkers of stress often drop within weeks. Blood pressure, cortisol, and inflammatory markers improve simply from breathing cleaner air or walking near trees. The environment constantly feeds the body with invisible nutrients: light of the right color, air of the right composition, silence of the right depth. Neglecting these inputs is like depriving cells of food. Longevity thrives where surroundings support rhythm. Daylight balanced by darkness, activity by rest, challenge by safety. To live long is to live in alignment with the planet's own cycles, its turning seasons, its daily rise and fall of light, its quiet intervals between storms. The environment is not a backdrop to life, it is a partner in endurance. When the partnership is respected, the conversation between organism and world continues for decades longer, gentle and clear, sustaining the biology that depends on it. Inside the human body lives an invisible world that rivals rainforests in complexity. Trillions of microbes, bacteria, fungi, archaea, and viruses inhabit the skin, mouth, lungs, and especially the gut. Collectively known as the microbiome, this community carries millions of genes, outnumbering our own genetic material by a wide margin. It influences digestion, immunity, metabolism, and even mood. Longevity depends on the harmony of this internal ecosystem. When balance holds, it supports repair and stability, but when disrupted, it quietly accelerates decline. In the gut, microbes process food molecules that human enzymes cannot handle alone. They break down fiber into short-chain fatty acids like butyrate, acetate, and propionate, which nourish intestinal cells and reduce inflammation. They synthesize vitamins such as K and B12, transform bile acids, and regulate how much energy we extract from food. A diverse microbiome acts as a flexible filter, adapting to changes in diet or environment while preserving equilibrium. Reduced diversity, by contrast, makes the system brittle, prone to overgrowth of harmful species and chronic immune activation. The immune system learns from these residents throughout life. In early childhood, exposure to microbes trains immune cells to distinguish between threat and tolerance. Without that education, defenses become jumpy, leading to allergies and autoimmune disease later on. In adults, microbial signals continue to shape immune tone. Molecules released by beneficial bacteria interact with receptors on intestinal and immune cells, dampening unnecessary inflammation. This crosstalk prevents the silent smoldering that underlies many age-related conditions. Diet is the main architect of microbial composition. Fiber from plants feeds beneficial species, while high sugar, processed fats, and alcohol favor those that thrive on excess. Fermented foods, yogurt, kimchi, kefir, sauerkraut, introduce live cultures that refresh diversity. Even small daily servings can alter microbial profiles within weeks. Polyphenols from fruits, teas, and herbs act as prebiotics, nourishing microbes that produce anti-inflammatory compounds. The longevity diets observed in Mediterranean and Asian regions naturally incorporate these foods, sustaining microbial richness across generations. Antibiotics, though life-saving, can disrupt this equilibrium. A single course may wipe out beneficial populations, leaving ecological gaps that take months to recover. Overuse in medicine and agriculture compounds the effect, selecting resistant strains that complicate human health. Restoration requires patience. Probiotics can help, but lasting recovery comes from consistent nourishment and exposure to natural environments where microbial diversity flourishes. Stress and sleep also reach the microbiome through hormonal and neural pathways. Cortisol, released under chronic tension, alters gut permeability and microbial balance. Poor sleep disrupts circadian rhythms that coordinate digestion, leading to erratic microbial activity. Meditation, regular rest, and physical movement indirectly nurture the gut by stabilizing the nervous system that governs it. This link forms the core of the gut-brain axis, a feedback loop in which microbes influence neurotransmitters such as serotonin and dopamine, shaping mood and cognition. The phrase gut feeling carries biological truth. Environmental contact plays a subtler role. People who garden, own pets, or spend time outdoors tend to harbor more diverse microbial communities. Soil bacteria and plant microbes refresh the skin and respiratory microbiomes, strengthening immunity. Sterile surroundings, while preventing infection, can starve the immune system of training opportunities. Balance again proves essential. Cleanliness without isolation. As the microbiome ages, beneficial species often decline while inflammatory ones expand. This shift mirrors changes in immune tone and metabolism, forming part of the wider pattern of biological aging. Emerging research shows that transplanting youthful microbiota into older animals rejuvenates aspects of their physiology, from improved insulin sensitivity to sharper cognition. Though such interventions remain experimental in humans, they highlight how intimately longevity depends on microscopic cooperation. To see the body as an ecosystem is to dissolve the boundary between self and environment. Every breath and meal invites new participants into our biology. Every heartbeat depends on their chemistry. Longevity, viewed this way, is not an individual achievement but a symphony of coexistence. Caring for this inner world, through diet, rhythm, calm, and connection with the outer world, sustains the foundation upon which all other repair depends. The more diverse and balanced this unseen forest becomes, the longer the host can thrive within it. Across the living world, there are creatures that defy decay by rebuilding what is lost. Their bodies blur the line between injury and renewal, offering glimpses into life's deepest capacity for repair. From salamanders regrowing limbs to planarian flatworms reconstructing entire bodies, these examples show that longevity is not only about slowing damage but about reversing it when possible. Regeneration is the purest form of maintenance, renewal elevated to an art. The process begins with cells that remember how to start over. When a salamander loses a leg, cells near the wound shed their specialized identities and revert to a stem-like state. This mass of versatile cells, called a blastema, begins dividing rapidly under the guidance of molecular signals that recreate the missing structure in perfect proportion. The nerves and blood vessels reconnect, bones reform, skin closes, and within weeks the limb functions as before. Genes once active during embryonic development awaken again, replaying the blueprint of growth. The animal's immune system cooperates, preventing scarring and allowing smooth regrowth, something mammals, including humans, rarely achieve. In simpler organisms, regeneration reaches astonishing extremes. Planarian flatworms can regrow an entire body from a fragment smaller than a grain of rice. Their stem cells, known as neoblasts, remain pluripotent throughout life, capable of producing every cell type indefinitely. If cut into pieces, each fragment becomes a complete new worm. Hydra, tiny freshwater polyps, display similar immortality through continuous cell turnover. Old cells slough off while new ones arise from a central reservoir of stem cells. Because this process never stops, hydra show no measurable aging. Their risk of death remains constant regardless of time. In these species, the fountain of youth is not myth but mechanism. Humans share many of the same genetic pathways that enable these feats, yet most are dialed down after development. Our stem cells are restricted to specific tissues, bone marrow, skin, intestine, liver, where they quietly maintain renewal but cannot rebuild whole organs. Evolution seems to have traded full regeneration for structural complexity and immune precision. Scarring, for instance, closes wounds quickly to prevent infection, but at the cost of perfect restoration. The balance between speed and fidelity shapes how different species age. Still, traces of deep regenerative potential remain. The human liver can regrow to its original size after major loss, guided by growth factors that synchronize cell division and vascular repair. Skin renews itself every few weeks, and the intestinal lining rebuilds completely every few days. Even in the heart and brain, long thought incapable of renewal, small populations of stem cells generate new cells throughout life, albeit at a slow rate. These sparks of regeneration hint that the capacity is not lost, only restrained. Researchers now explore how to awaken these dormant abilities safely. Techniques involving stem cell therapy, tissue engineering, and genetic reprogramming aim to coax mature cells back into youthful states. The discovery of Yamanaka factors, a small set of genes that reset cell identity, has opened paths to partial rejuvenation. In laboratory animals, carefully timed activation of these factors reverses some markers of aging without erasing cellular memory. The challenge lies in control. Complete reprogramming can produce tumors instead of tissue. Longevity science therefore seeks moderation, as always, restoring flexibility without losing stability. Nature offers other insights. Some species, like the axolotl, delay metamorphosis indefinitely, remaining in a larval state with lifelong regeneration. Others, such as sea cucumbers, expel internal organs when threatened and rebuild them weeks later. These strategies illustrate that renewal and defense can coexist, that survival sometimes means rebuilding from within rather than resisting from without. Each represents a unique solution to the same universal equation: how to preserve function through time. For humans, studying these creatures does more than promise medical breakthroughs. It reframes aging itself. Regeneration is not foreign to us but foundational. Our bodies constantly replace, recycle, and remodel, even if we rarely notice. Every new layer of skin, every repaired strand of DNA, is a quiet echo of the salamander's limb and the hydra's endless renewal. Longevity, viewed through this lens, is simply the continuation of regeneration at every scale. The more faithfully we support that renewal, through rest, nutrition, and avoidance of chronic damage, the longer the pattern can repeat. Life endures not because it avoids injury, but because it remembers how to begin again. Every cell in the human body carries the same DNA, yet a skin cell looks nothing like a neuron or a liver cell. The difference lies not in the genetic script itself, but in how it is read. The system that manages this reading is called epigenetics, a layer of chemical markings that instructs genes when to turn on, when to stay silent, and how strongly to express themselves. This system allows identical genetic code to produce astonishing diversity and gives life the flexibility to adapt to its surroundings without rewriting its blueprint. Over time, however, this epigenetic language drifts, and with it, the accuracy of cellular identity fades. Longevity depends on how clearly the genome can continue to read its own instructions. Epigenetic regulation works mainly through two mechanisms: DNA methylation and histone modification. Methyl groups attach to specific bases in DNA, acting as punctuation marks that silence certain genes. Histones, the proteins around which DNA coils, can be tagged with chemical groups that tighten or loosen their grip, altering gene accessibility. These modifications respond to signals from hormones, nutrients, toxins, and stress. When conditions change, the pattern of marks changes too, enabling rapid adaptation without altering DNA sequence. This adaptability is the biological equivalent of learning. But memory, whether genetic or neural, is vulnerable to noise. As cells divide, their epigenetic marks must be copied along with DNA. Errors accumulate. Some genes remain active when they should rest, others fall silent when needed. Over decades, these misprints disrupt coordination between repair, metabolism, and inflammation. Researchers can now estimate biological age by analyzing methylation patterns across the genome. These so-called epigenetic clocks often predict health outcomes more accurately than chronological years, revealing how environment and lifestyle accelerate or decelerate molecular aging. Unlike fixed mutations, epigenetic marks are reversible. This reversibility has made them a focus of modern longevity research. Diet, exercise, stress management, and sleep all reshape the epigenome daily. Physical activity increases beneficial methylation of genes involved in metabolism and inflammation control. Nutrients such as folate, choline, and B vitamins supply the chemical groups used in methylation, ensuring that repair instructions remain clear. Periods of fasting and caloric moderation trigger histone changes that favor maintenance pathways over growth. Even meditation and social connection influence stress-related hormones that indirectly tune epigenetic expression. The genome listens constantly to its environment. Longevity arises from giving it steady, constructive messages. At the frontier of science, researchers experiment with epigenetic reprogramming, turning back cellular age by partially resetting these marks. The discovery of 4 key genes, known as Yamanaka factors, showed that mature cells can revert to a stem-like state capable of becoming any tissue. Applied too completely, the process erases identity, but applied briefly, it appears to rejuvenate without losing memory. In mice, cyclic activation of these factors restores youthful patterns of gene expression, improves organ function, and extends lifespan. Translating such results to humans requires caution, yet the principle is clear. Aging is not only accumulation of damage but also confusion in regulation, clarifying that regulation can restore coherence. Epigenetics also explains how experience passes between generations. Stress, nutrition, and toxins can alter parental methylation patterns and influence offspring development, even without changing DNA. This transgenerational inheritance extends the reach of behavior far beyond a single lifetime. It also implies that nurturing environments today can plant resilience in future generations. Longevity, then, is not solely an individual pursuit but a continuing lineage of molecular education. The concept reframes the relationship between genes and choice. DNA provides potential. Epigenetics interprets it through behavior. Lifestyle becomes a form of authorship, editing the margins of the genome with every decision. The same sequence that once predicted vulnerability can express strength under different conditions. This plasticity is both humbling and empowering. We inherit tendencies, not destinies. In practical terms, supporting the epigenome means aligning daily rhythm with natural cycles, consistent sleep, daylight exposure, whole foods, movement, and calm. These behaviors keep the cellular script legible, preventing the drift that clouds communication with age. On a deeper level, the lesson is philosophical. Identity in biology, as in life, endures not through rigidity but through responsive balance. Cells, like people, remain young when they remember how to adapt without forgetting who they are. Among all interventions ever studied in biology, caloric restriction stands out as one of the most consistent in extending lifespan across species. From yeast and worms to rodents and primates, reducing total calorie intake without malnutrition slows aging, delays disease, and improves resilience. The discovery reshaped how scientists understand metabolism. Longevity is not only about what we eat, but about how much energy the body must process to stay alive. The first formal observation came in the 1930s, when researchers at Cornell University found that mice fed less lived up to 40% longer than their well-fed counterparts. Their coats stayed sleek, their activity higher, their organs healthier. Since then, the effect has been confirmed in countless studies, revealing that moderate scarcity activates survival programs deep within cells. In times of famine, life evolved to endure by shifting from growth to preservation, recycling damaged components, and improving efficiency until food returned. Caloric restriction reproduces that signal of scarcity without true starvation. The molecular players behind this adaptation form a familiar network. Low nutrient intake suppresses the mTOR pathway, reducing protein synthesis and growth signals. Simultaneously, it activates AMP-activated protein kinase, AMPK, which senses low cellular energy and promotes repair and autophagy. Sirtuins, enzymes that depend on NADMYs, become more active, coordinating DNA maintenance and mitochondrial function. Together, these shifts push the cell into a protective state, conserving energy while clearing waste. The process resembles a factory temporarily shutting down production lines to service its machinery. Autophagy, literally self-eating, is central to this renewal. During restricted intake, cells dismantle worn-out proteins, membranes, and organelles, recycling their components for new use. This reduces the buildup of cellular debris that contributes to inflammation and degeneration. When normal feeding resumes, the rejuvenated machinery operates with greater precision. Intermittent fasting, which alternates periods of eating and abstention, appears to stimulate similar mechanisms while remaining more practical for humans than continuous restriction. Not all organisms respond equally. The degree of benefit depends on species, genetics, and environment. In short-lived animals, the impact is dramatic because metabolism shifts rapidly. In long-lived primates, including humans, effects accumulate slowly but still influence biomarkers of aging, lower insulin levels, improved lipid profiles, reduced oxidative stress, and sharper cognitive performance. Studies on rhesus monkeys show fewer age-related diseases and longer health span, even if maximum lifespan increases modestly. The primary advantage seems to be quality of life rather than sheer duration. Humans practicing moderate caloric restriction often display metabolic profiles resembling those of younger individuals: reduced inflammation, better blood pressure, improved insulin sensitivity, and favorable gene expression related to stress resistance. Yet total deprivation can backfire, leading to muscle loss, hormonal imbalance, or psychological strain. The goal is not hunger but metabolic balance, eating slightly less than abundance demands, with nutrient density high enough to meet all essential needs. Diets rich in plants, moderate in protein, and low in processed sugars achieve this balance naturally. Cultural traditions anticipated these findings long before laboratories did. Religious fasting, seasonal scarcity, and mindful restraint appear in nearly every civilization's history. These practices not only preserved resources but allowed the body to reset. The rhythm of feast and pause mirrors ecological cycles of plenty and rest. Modern abundance, with constant access to food, removes those intervals, keeping growth signals permanently switched on. Reintroducing brief, structured scarcity restores the alternation evolution expects. Mechanistically, caloric restriction also influences the epigenome, the chemical markings on DNA that regulate gene activity. Reduced nutrient flow recalibrates methylation patterns, silencing pro-inflammatory genes and reactivating maintenance pathways. This may partly explain why even short periods of fasting yield benefits disproportionate to their length. The body interprets scarcity as an opportunity to refine its operations rather than a threat to survival. Psychologically, moderate restriction cultivates awareness. It reminds the organism and the mind that consumption has a rhythm. Hunger, when mild and deliberate, sharpens perception and gratitude, aligning internal focus with biological clarity. For those pursuing longevity, the lesson is not deprivation but discipline, giving the body time to clean, recalibrate, and rest between cycles of abundance. Caloric restriction proves that the biology of aging listens to rhythm more than to abundance. Energy intake, like all forms of stress, must fluctuate. Within that fluctuation lies renewal. When scarcity is brief, balanced, and intentional, the organism responds not with weakness but with efficiency, strengthening the very systems that keep life stable across time. Movement is the most ancient medicine. Long before humans named exercises or invented sports, motion defined life itself. From the single-celled organism drifting toward light to the heartbeat pulsing within a mammal's chest, activity marks the difference between existence and inertia. Longevity thrives not through stillness but through rhythm, steady, balanced movement that keeps every system in conversation with the rest. Biologically, motion maintains structure. Muscles contract and relax, pumping blood and lymph through the body. This circulation delivers oxygen and nutrients while removing waste products that would otherwise accumulate. Without regular motion, cells drown in their own byproducts, and tissues lose elasticity. Even gentle movement, such as walking, stretches connective fibers, lubricates joints, and keeps mitochondria alert. Studies show that sitting for long hours suppresses genes related to glucose metabolism within minutes, demonstrating how quickly the body notices immobility. At the molecular level, exercise triggers a cascade of signals that echo those of caloric moderation and hormetic stress. During activity, energy demand rises. AMP-activated protein kinase, AMPK, senses the shortage and promotes efficiency, increasing mitochondrial number and improving insulin sensitivity. Simultaneously, mild oxidative stress activates antioxidant defenses and repair enzymes. Over time, this adaptation builds resilience, The body learns to handle stress with smaller inflammatory responses and faster recovery, precisely the pattern seen in long-lived species. Different forms of movement train different systems of longevity. Aerobic activity, such as walking, swimming, or cycling, enhances cardiovascular endurance and stimulates neurogenesis in the hippocampus, preserving memory. Strength training maintains muscle mass and bone density, counteracting the natural decline of growth hormone and testosterone. Flexibility and balance exercises, like yoga or Tai Chi, support posture, coordination, and calm parasympathetic activity. Together, they form a complete language of motion: endurance for the heart, stability for the frame, and awareness for the mind. The relationship between movement and brain health is especially intimate. Each contraction of muscle releases proteins called myokines into the bloodstream. These molecules travel to the brain, where they reduce inflammation and encourage the growth of new neural connections. Physical activity also increases blood flow and oxygen delivery to the cortex, sharpening attention and mood. In this sense, exercise acts as both prevention and therapy, protecting cognition while soothing emotional turbulence. Regular activity influences the endocrine system as well. It lowers baseline cortisol, balances insulin, and improves circadian alignment. Morning movement synchronizes the body clock, while light evening stretching promotes calm. The timing of activity reinforces the internal rhythm that governs hormone release and metabolism. When movement follows the cycle of daylight and rest, physiology finds its natural pulse again. Longevity depends less on intensity than on consistency. Sudden bursts of effort followed by long inactivity create mechanical and metabolic strain. In contrast, moderate daily motion sustains the benefits without exhaustion. Populations known for exceptional lifespan, from Okinawa to Sardinia, rarely engage in formal exercise. Instead, movement is woven naturally into daily life: walking to neighbors, tending gardens, carrying food, kneeling, rising. This constant, low-level activity keeps muscles toned and circulation open without overwhelming repair systems. Modern environments challenge this rhythm. Machines move for us, chairs shape our days. To restore equilibrium, movement must be intentional again. Even short intervals, standing, stretching, taking stairs, breathing deeply, reset metabolic sensors and reawaken dormant circulation. The key is frequency, not heroics. The body values repetition more than intensity. Each gentle signal reminds it to remain alive in motion. Recovery completes the cycle. Muscles grow stronger not during exertion but in rest, when repair pathways rebuild fibers. Alternating activity with adequate sleep and nutrition turns exercise from stress into renewal. Without recovery, even healthy motion becomes destructive. As with all aspects of longevity, rhythm, proportion, and patience define success. Movement unites the physical and the mental. It grounds thought in sensation and reminds the organism of its design: to move, adapt, and respond. The same principles that preserve muscle also preserve identity. Use what you have, renew it often, and keep it flexible. A body that moves daily teaches its cells that life continues, that renewal remains possible. Longevity, then, is not a still achievement but a choreography, repeated each day in every step, stretch, and breath that carries us forward. Every process that sustains life requires pauses. Muscles need time to rebuild after movement, neurons to reset after thought, and hormones to realign after stress. Longevity is not achieved by continuous effort but by the alternation of activity and restoration. Rest is the second half of every function, the silent phase that gives meaning to motion. At the cellular level, recovery means repair. After exertion or stress, cells accumulate microdamage, broken proteins, misfolded enzymes, and fragments of oxidized molecules. During rest, repair enzymes scan DNA for errors, mitochondria recycle damaged components, and antioxidant systems neutralize leftover reactive oxygen species. The process is energy-intensive, which is why it happens most efficiently when the body is quiet and metabolism slows. Without these nightly interventions, errors accumulate, and tissues lose their fine-tuning. Sleep is the most visible form of recovery. While consciousness dims, the body engages in its most complex maintenance program. Growth hormone peaks, directing protein synthesis and tissue rebuilding. The glymphatic system flushes metabolic waste from the brain, clearing debris that, if left unchecked, contributes to cognitive decline. Memory traces consolidate as neural circuits replay the day's experiences, strengthening useful connections and pruning excess. These invisible chores explain why even a few nights of insufficient sleep raise inflammation, blunt focus, and disturb metabolism. Restoration is not a luxury, it is the hidden half of survival. Beyond nightly rest, periodic withdrawal from constant stimulation serves a similar role. Ancient traditions wove this need into culture through Sabbath days, seasonal retreats, or winter dormancy. Modern life erodes these intervals with perpetual light and information. The nervous system, designed for pulses of challenge and calm, remains half alert, producing low-grade fatigue that shortens health span. Reintroducing quiet moments, reading, stillness, slow breathing, restores parasympathetic tone, lowering heart rate and blood pressure while enhancing immune coordination. These practices re-establish the biological rhythm that evolution assumed. The chemistry of rest is measurable. When the parasympathetic system dominates, cortisol falls and insulin sensitivity improves. In this state, digestion deepens and nutrient absorption increases, feeding the very repair processes that preserve youth. Deep breathing expands lung capacity, raises oxygen saturation, and signals safety to the brainstem. Meditation alters electrical patterns in the cortex, shifting waves toward alpha and theta frequencies associated with integration and creativity. The border between body and mind dissolves here. Physiological rest and psychological calm reinforce each other. Recovery from exercise follows the same pattern of balance. After strain, microtears in muscle fibers attract immune cells that initiate healing. If rest and nutrition are sufficient, new fibers form stronger than before. If rest is skipped, inflammation lingers, weakening structure instead of building it. The difference between training and overtraining is recovery time. Even elite athletes extend longevity, not through intensity alone, but through disciplined restoration, sleep, hydration, and controlled intervals of stillness. On a broader scale, recovery applies to the senses and the mind. Continuous noise, light, or digital stimulation keep neural circuits firing long past necessity. Vision and hearing adapt to variety. Constant exposure dulls them. Periods of sensory quiet recalibrate thresholds, allowing subtle perception to return. Mental rest, disengaging from problem-solving or planning, restores creativity by letting unconscious networks reorganize information. Many insights arrive not during concentration but during pause, when the brain drifts freely between associations. The immune system also listens to rest. During relaxation, lymph flow increases, circulating white blood cells more effectively. Fever and wound healing accelerate during sleep because energy is diverted from activity to repair. Even the microbiome shifts composition during restful hours, optimizing digestion and detoxification. The pattern repeats across scales. Calm supports order, exhaustion invites entropy. Culturally, societies that respect rest often outlive those that glorify constant activity. Afternoon breaks, communal meals, and unhurried evenings reflect biological wisdom rather than laziness. They enforce cycles that modern schedules ignore. Longevity arises where human rhythm still echoes natural rhythm, sunrise to sunset, effort to ease, inhalation to exhalation. In the physics of life, every action requires recovery to maintain equilibrium. Without pause, systems overheat. Without challenge, they stagnate. The art of long living is therefore the art of pacing, working with precision, then stepping back to let restoration unfold. Within each pause, the body rewrites its structure, the mind regains clarity, and time itself seems to slow. Rest does not interrupt progress, it sustains it, extending the arc of vitality far into the years ahead. Every second, trillions of chemical reactions unfold within the human body. Most occur flawlessly, but some go astray, breaking bonds, distorting structures, or leaving fragments behind. This microscopic wear accumulates over time, forming the molecular basis of aging. Longevity, at its core, depends on how efficiently organisms detect and repair these errors before they cascade into dysfunction. The quiet diligence of molecular maintenance determines whether life endures with grace or frays at the edges. DNA, the most critical molecule of all, endures constant assault. Ultraviolet light, reactive oxygen species, and even natural replication errors strike the genome millions of times per day. To counter this, cells deploy overlapping repair systems. Base excision repair corrects single-letter mistakes caused by oxidation. Nucleotide excision repair removes larger lesions, such as those left by ultraviolet exposure. Mismatch repair scans freshly copied DNA for typos that escaped proofreading. When damage overwhelms these systems, double-strand break repair, via homologous recombination or non-homologous end joining, reconstructs severed chromosomes. These mechanisms function like editors and restorers, preserving coherence through continuous revision. Failures in any step can trigger mutations, cancer, or cell death. Proteins face a different threat: structural deformation. Oxidative stress and glycation twist their three-dimensional shapes, impairing function. The body relies on proteostasis, a network of chaperone proteins, proteasomes, and autophagy pathways, to manage quality control. Chaperones refold misshapen proteins. Proteasomes disassemble those beyond repair. Autophagy engulfs entire damaged organelles for recycling. This triage keeps the intracellular environment clean and efficient. When proteostasis falters with age, damaged proteins aggregate, as seen in Alzheimer's and Parkinson's disease. Maintaining this equilibrium is one of longevity's most decisive challenges. Lipids and cellular membranes also deteriorate. Polyunsaturated fats oxidize easily, compromising the flexibility and signaling functions of membranes. Antioxidants, both enzymatic, such as superoxide dismutase and glutathione peroxidase, and dietary, such as vitamin E, neutralize radicals before they propagate chain reactions. Repair enzymes then remodel membranes by replacing damaged lipids. These invisible corrections occur billions of times daily, sustaining the integrity that cells depend on for communication. Mitochondria, the cell's power plants, generate energy but also the majority of reactive oxygen species. Their own circular DNA is especially vulnerable because it lacks the protective histones of nuclear DNA. When mitochondria sustain damage, they can trigger inflammation or cell death. To counter this, cells practice mitophagy, selectively dismantling defective mitochondria and replacing them with new ones through biogenesis. Exercise, fasting, and thermal stress all enhance this turnover, ensuring a younger, more efficient mitochondrial population, a clear link between behavior and molecular renewal. At larger scales, damaged cells are eliminated through apoptosis, a programmed form of self-destruction that prevents harm to neighboring tissue. In young organisms, apoptosis clears space for regeneration. In older ones, it must balance removal with preservation. When apoptosis stalls, senescent cells accumulate. These aged cells no longer divide but secrete inflammatory molecules, creating a toxic environment known as the senescence-associated secretory phenotype. Modern therapies called senolytics aim to remove these lingering cells, restoring tissue function and reducing systemic inflammation. In animal studies, periodic clearance of senescent cells improves organ performance and extends healthy lifespan. Environmental influences amplify or reduce the load of molecular damage. Radiation, pollution, tobacco smoke, and excessive calories all increase oxidative stress. Protective compounds from plants, polyphenols, carotenoids, sulfur compounds, stimulate detoxification enzymes that neutralize reactive species. This hormetic activation mirrors the body's natural defenses, training them to respond quickly when true threats appear. The same logic applies to mild physical challenges, such as cold or exercise. Short bursts of stress strengthen repair efficiency by keeping these pathways active. Ultimately, Molecular maintenance resembles housekeeping at a scale too small to see. Each night, cells collect waste, patch broken molecules, and replace what can no longer be fixed. The process never ends. It is life itself. When the repair crews slow, entropy gains ground. Longevity arises from sustaining their pace through balanced metabolism, nutrient sufficiency, and controlled exposure to stress. Every heartbeat, every breath, carries with it an invisible orchestra of restoration that rewrites the body's structure moment by moment. The art of living long is the art of giving those mechanisms time, energy, and reason to continue their work. No two human beings age in quite the same way. Even among people who live similar lives, eat the same foods, and share the same environment, some reach advanced age with strength intact while others decline early. The difference often lies in subtle variations within their DNA, small genetic differences that shape metabolism, repair, and resilience. Longevity is therefore both universal and personal, bound by shared biology, yet fine-tuned by inherited diversity. Scientists study families and populations where exceptional age clusters, searching for patterns. In places such as Sardinia, Okinawa, and Ikaria, certain lineages show a higher frequency of centenarians. Their diets and lifestyles play major roles, but genetics add another layer of defense. Variants in genes related to inflammation, lipid metabolism, and cellular repair appear more common among these individuals. For instance, versions of the FOXO3A gene, which encodes a transcription factor governing stress resistance and insulin signaling, are strongly associated with longer lifespan. This gene helps cells respond calmly to oxidative stress, promoting maintenance rather than panic. Carriers of favorable FOXO3A variants often display better cardiovascular health and slower biological aging. Another set of genes influences lipid processing. People with particular variants of APOE, especially the APOE2 type, tend to have lower risk of neurodegenerative disease and longer lives. APOE governs cholesterol transport and brain repair. Its less fortunate counterpart, APOE4, increases inflammation and vulnerability to Alzheimer's disease. These differences highlight how longevity is not controlled by a single longevity gene, but by the harmony or discord among many small effects. Repair efficiency is another genetically guided trait. Variations in genes such as SIRT1, WRN, and LMNA, involved in DNA stability and chromosomal maintenance, determine how quickly damage accumulates. Rare mutations in these genes can cause premature aging syndromes, revealing how crucial their normal function is for everyday endurance. Conversely, naturally efficient versions of the same genes appear to grant extra decades of vitality. The lesson is that aging and longevity lie on a continuum defined by repair capacity. The slower the damage, the longer the grace. Immune genes also shape lifespan. The HLA complex, which determines how the body recognizes foreign antigens, varies widely among individuals. Balanced variants help the immune system respond precisely without overreacting. Those that tilt toward chronic activation raise the risk of autoimmune disorders and inflammation, major drivers of aging. Centenarians often exhibit immune profiles marked by moderation, strong defense, rapid resolution, minimal collateral damage. Yet genes explain only part of the story. Identical twins, sharing nearly identical DNA, often diverge significantly in lifespan due to environmental and epigenetic influences. Lifestyle modifies how genes express themselves. Nutrition, sleep, stress, and exposure all alter the molecular environment in which genes operate. In this way, inheritance sets boundaries, but behavior fills the space between them. The flexibility of epigenetics ensures that even modest choices can compensate for genetic disadvantage or enhance existing strengths. Population studies underscore the interaction between genetics and culture. The same gene variant may help or hinder depending on context. A metabolism tuned for scarcity may protect against starvation but promote obesity in abundance. Genes that heighten inflammation may defend against infection in youth yet cause tissue damage later. Longevity arises where genetic programming and modern environment remain compatible, where inherited strategies still match daily conditions. Advances in genomics now allow people to glimpse their personal longevity blueprint. Whole genome sequencing identifies variants that influence aging pathways, metabolism, and disease risk. While tempting to view these data as destiny, the reality is more nuanced. Genes suggest probabilities, not guarantees. A predisposition toward longer life must still be nurtured through choices that support repair and balance. Conversely, unfavorable variants can often be offset by habits that reduce stress, maintain cardiovascular health, and protect DNA integrity. The interplay between nature and nurture remains fluid to the very end. In the broader picture, genetic diversity ensures humanity's survival as a species. Some lineages thrive in cold, others in heat. Some resist infection, others adapt to scarcity. Evolution preserves this variety because longevity, like all traits, benefits from flexibility. The genes that let a few live beyond a century may not help in every environment, but they expand the spectrum of possibilities. Within that spectrum, each person holds a unique composition of resilience, a molecular individuality that, when harmonized with environment and habit, defines the length and quality of life. Every tissue, every organ, and every thought originates from the cell. These microscopic units are not mere building blocks, but self-contained ecosystems, complete laboratories of chemistry, communication, and adaptation. Understanding longevity ultimately means understanding how cells preserve order while surrounded by constant change. When the cell remains balanced, the organism endures. When its internal systems drift out of coordination, the entire structure begins to age. A healthy cell maintains three pillars of stability: energy balance, structural integrity, and communication. Energy comes from mitochondria, which convert nutrients into adenosine triphosphate, ATP, the universal fuel of life. Structural integrity depends on the cytoskeleton, a dynamic network of filaments that gives shape, transports materials, and anchors essential enzymes. Communication occurs through signaling molecules that tell the cell when to divide, when to repair, and when to rest. All three functions are synchronized by feedback loops that evolve to resist entropy. Longevity is, in essence, the persistence of these loops through countless cycles of wear and repair. At the biochemical level, the cell's environment is tightly regulated. Inside, enzymes work best within narrow ranges of temperature, pH, and ionic balance. The cell membrane acts as both barrier and gatekeeper, controlling what enters and leaves. Lipid layers and embedded proteins ensure selective permeability, allowing nutrients in and waste out. When this membrane stiffens with age, often due to oxidation or loss of unsaturated fats, communication falters. Nutrient transport slows, receptors misfire, and energy efficiency drops. Maintaining membrane flexibility through balanced nutrition and antioxidants supports cellular youth at its most fundamental layer. DNA lies in the nucleus like a library, yet it is the RNA and protein synthesis machinery that determines how the library's books are read. Ribosomes translate genetic code into proteins that perform every task from metabolism to defense. Errors in this translation process rise with age, producing defective proteins that stress the repair systems. The cell's ability to detect and discard these mistakes defines its health span. Caloric balance, moderate stress, and sleep all assist translation accuracy by providing sufficient energy and hormonal alignment for quality control. Communication between cells forms the next layer of resilience. Neighboring cells exchange ions, hormones, and growth factors, creating tissue-wide coherence. This coordination depends on gap junctions and chemical gradients that decline with chronic inflammation or oxidative stress. When messages become distorted, tissues lose harmony, heart rhythms waver, wound healing slows, and immune reactions overshoot. Restoring intercellular clarity through hydration, nutrient balance, and stress reduction allows signals to travel cleanly again, preventing the miscommunication that accelerates biological aging. Another essential factor is cellular turnover, the balance between division and death. Stem cells supply replacements as old or damaged cells retire through apoptosis. In youth, This cycle runs smoothly, but with age, stem cells tire and divisions become error-prone. Supporting their vitality through consistent blood flow, adequate sleep, and anti-inflammatory nutrition preserves the renewal engine of the body. In experimental studies, youthful circulation, rich in growth factors and oxygen, can rejuvenate aged stem cells, suggesting that systemic health feeds cellular youth directly. The cell also senses its surroundings through mechanical and chemical cues, a process called mechanotransduction. Muscles, bones, and connective tissue constantly transmit small mechanical forces that tell cells how strong to build, how dense to become, or when to rest. Regular movement keeps these signals alive. Inactivity silences them, leading to loss of structure. Thus, even at the microscopic scale, exercise functions as conversation, reminding cells that their architecture still matters. When cells struggle, the body does not fail immediately. It compensates, borrowing capacity from neighbors or organs. But over decades, the cumulative loss of cellular precision turns subtle inefficiencies into visible aging. The remedy lies not in replacing every cell, but in maintaining the environment in which they can thrive. A landscape of balanced nutrients, clean water, regular movement, restorative sleep, and calm neural tone. Longevity, therefore, is cellular coherence scaled to the size of a human being. Each heartbeat represents billions of synchronized reactions within countless microscopic laboratories. When those laboratories remain orderly, life feels effortless. The secret is not immortality but maintenance, the quiet, continuous care that allows each cell to remember its purpose and carry it out faithfully for as long as possible. Longevity depends as much on coordination as on repair. A body can possess strong organs and efficient cells, yet still decline if its internal communication falters. From hormones to electrical impulses, the human organism is sustained by an intricate web of messages that travel with perfect timing and clarity. When those signals stay synchronized, tissues cooperate like instruments in an orchestra. When they drift out of rhythm, aging begins to sound like static. Communication starts with the nervous system, the body's fastest messenger. Electrical impulses travel through neurons at speeds of up to 100 meters per second, carrying information from brain to muscle, from organ to organ. Each signal ends in a tiny chemical conversation called a synapse, where neurotransmitters leap across microscopic gaps to continue the message. Over time, these circuits must remain both stable and flexible. Too rigid, and the body cannot adapt. Too loose, and coordination dissolves. Sleep, nutrition, and constant mental engagement keep these connections strong, ensuring that the brain's intentions reach every cell with precision. The autonomic nervous system manages what we rarely notice: heartbeat, digestion, temperature, and breathing. It balances between two complementary forces. The sympathetic branch accelerates, preparing the body for action. The parasympathetic branch slows, guiding recovery and repair. Longevity arises when these forces alternate smoothly, effort followed by ease, alertness followed by calm. Modern stress keeps the accelerator pressed, leaving little room for restoration. Practices that lengthen exhalation, slow the heart, or quiet thought allow parasympathetic tone to return, restoring equilibrium across the network. Chemical communication overlays the electrical one. Hormones, secreted into the bloodstream, act as slower but more global messages. A single pulse of insulin or cortisol can influence thousands of genes in distant tissues. Timing and dosage matter. Too frequent, and sensitivity fades. Too sparse, and coordination weakens. The decline of hormonal rhythm with age explains much of what we call aging: fatigue, disrupted sleep, slower repair. Yet small adjustments in lifestyle can resynchronize these rhythms. Regular light exposure resets melatonin cycles, moderate fasting stabilizes insulin, and consistent movement maintains growth hormone pulses. Every cell also communicates locally. Chemical gradients, calcium waves, and molecular vibrations link neighboring cells within tissues. These exchanges govern growth, inflammation, and regeneration. For example, when skin is injured, Damaged cells release signaling molecules that summon fibroblasts to rebuild collagen and blood vessels. When the repair is done, another signal instructs them to stop. If that second message fails, scarring results. Aging often reflects such incomplete signaling. The conversation continues too long or stops too soon. The vascular system adds a physical dimension to communication. Blood vessels deliver not only oxygen and nutrients, but also molecular messages from one organ to another. The endothelium, the inner lining of every vessel, releases nitric oxide, a gas that widens arteries and coordinates flow. Inflammation, high blood sugar, and lack of movement stiffen this lining, reducing responsiveness. Maintaining vascular flexibility through regular activity, hydration, and balanced nutrition ensures that messages carried by blood reach their destinations swiftly and clearly. A newer discovery reveals that even organs once thought independent send biochemical notes to each other. The gut communicates with the brain through hormones and nerves. Fat tissue releases adipokines that influence appetite and immunity. Skeletal muscles send myokines that affect cognition and repair. The body is not a collection of parts, but a single talking system. Each organ speaks its dialect, and health depends on the harmony of their dialogue. At the molecular edge, communication involves extracellular vesicles, tiny bubbles that shuttle RNA and proteins between cells. They act like couriers, transmitting repair instructions or distress calls. Research suggests that young cells send vesicles rich in protective molecules, while aging cells release signals that propagate decline. Therapies that enhance beneficial vesicle exchange may one day slow or even reverse some aspects of aging by restoring clarity to this microscopic conversation. When communication weakens, coordination gives way to noise. Misdirected hormones, errant electrical rhythms, chronic inflammation. The cure is not silence but clarity, allowing each signal its proper place and time. Good sleep, clean air, sunlight, moderate challenge, and emotional calm all contribute to this clarity by aligning biological clocks and stabilizing chemistry. Life, viewed through communication, is a continuous broadcast between trillions of participants. Each heartbeat, thought, and breath depends on the accuracy of that broadcast. Longevity is the art of keeping the channels open, listening, responding, and returning to balance. When the body speaks clearly to itself, it knows how to endure. All living systems face a fundamental decision, whether to grow or to maintain. Growth expands structure, building new cells, tissues, and complexity, while maintenance preserves what already exists. In early life, growth dominates, energy pours into construction. As maturity approaches, the balance must shift toward preservation, or the machinery will overheat and wear itself out. Longevity depends on how gracefully that shift occurs and how consistently the body can favor maintenance without losing vitality. In the young organism, growth is relentless. Hormones such as growth hormone, insulin, and IGF once surge through the bloodstream, driving rapid cell division and protein synthesis. These processes consume enormous energy and generate oxidative byproducts that accumulate over time. Evolution designed this high-speed phase to ensure survival until reproduction. Beyond that, the imperative weakens. Yet in modern life, abundant food and constant stimulation keep growth signals active long after their evolutionary purpose has passed. Cells remain in a mild state of overdrive, prioritizing production over repair. The result is early exhaustion of molecular machinery. Maintenance follows a quieter logic. It invests energy not in expansion but in precision. Pathways such as AMP-activated protein kinase and sirtuins sense low nutrient states and direct resources toward repair. DNA correction, protein recycling, and mitochondrial renewal. The mTOR pathway, which governs cell growth, must therefore be modulated, not silenced. When mTOR slows, autophagy awakens, the cleaning mechanism that clears out broken components. This shift from construction to conservation marks the transition from youth to sustainable life. Organisms that master this transition live longest. Tortoises, whales, and bats all display slow growth followed by efficient maintenance. Their metabolic engines idle rather than roar, avoiding unnecessary damage. Human biology follows the same principle. Slowing growth does not mean stagnation but refinement. Moderate caloric intake, consistent activity, and periods of fasting naturally tilt, signaling toward preservation, allowing tissues to renew without the strain of continuous expansion. The balance between growth and maintenance also plays out within tissues. Muscles, for instance, need enough protein synthesis to stay strong but also enough rest to repair micro-tears. Bones remodel constantly, breaking down old mineral and replacing it with new. Even neurons, though largely non-dividing, must maintain synaptic plasticity while preventing excitatory overload. Each of these systems survives through rhythm, alternating building and restoration in steady cadence. Overemphasizing one side disrupts the other. Excessive growth without maintenance leads to fragility, while excessive conservation without activity leads to decline. Hormones translate this balance into daily cycles. Cortisol and adrenaline promote readiness and growth in the morning. Melatonin and growth hormone orchestrate repair during sleep. Disrupting this rhythm through erratic schedules or artificial light confuses the body's allocation of resources. Aligning activity with daylight and rest with darkness ensures that energy flows naturally between creation and conservation. This synchronization may be one of the simplest and most powerful tools for extending health span. Psychologically, the same principle applies. Human ambition fuels creation, career, family, knowledge, but constant pursuit without reflection leads to burnout. Just as cells must pause to repair, minds need intervals of quiet to integrate experience. Reflection is cognitive maintenance. It consolidates learning, clears emotional residue, and prevents the accumulation of mental noise. Cultures that honor both work and contemplation, effort and stillness, mirror the biological rhythm that longevity requires. Modern medicine increasingly targets this duality. Drugs like rapamycin, which inhibit mTOR, and compounds that activate AMPK or sirtuins, aim to mimic the metabolic signature of maintenance without severe restriction. Early studies suggest they can delay age-related disease, though the long-term effects remain under study. Still, they confirm what evolution already encoded. The same pathways that drive growth can, when slowed, extend survival. Ultimately, longevity is not about suppressing vitality but directing it wisely. Growth builds the form, maintenance preserves its function. Every heartbeat and breath balance these two imperatives. When the body learns to alternate between construction and care, between ambition and rest, it stops wasting energy fighting itself. The secret is proportion. To build just enough, to rest just enough, and to allow renewal to follow creation as naturally as night follows day. That rhythm, repeated quietly through the decades, is the mathematics of endurance. Sleep is not a pause in life, it is the hidden engine of preservation. During those hours of stillness, the body reorganizes itself, restoring order where daytime activity has left chaos. For longevity, sleep is both foundation and compass, resetting the rhythm that every cell depends on to know when to grow, when to repair, and when simply to rest. The human sleep cycle alternates between two primary states, non-REM and REM. Non-REM sleep is divided into light, intermediate, and deep stages. In deep sleep, growth hormone surges, stimulating protein synthesis and tissue repair. Cells clear damaged proteins and replace exhausted molecules. Heart rate and blood pressure drop, conserving energy and reducing mechanical stress on vessels. In REM sleep, the brain becomes active again, replaying the day's neural patterns to strengthen memory and learning. Together, these phases create a nightly pattern of physical renewal and cognitive refinement, an internal maintenance program run by the circadian clock. This clock, located in the suprachiasmatic nucleus of the hypothalamus, responds primarily to light. Morning sunlight synchronizes it. Darkness allows melatonin to rise and signal the body that night has arrived. Artificial lighting, late screens, and irregular schedules confuse the system, delaying melatonin release and fragmenting deep sleep. Over years, such disruption increases risk of metabolic disease, depression, and cardiovascular strain. Maintaining consistent sleep and wake times anchors the body's entire hormonal rhythm, aligning metabolism, temperature, and immune function. Sleep is also when the brain cleans itself. During waking hours, neurons release waste molecules into the interstitial space. At night, that space expands by nearly 60% as cerebrospinal fluid flows through, flushing toxins such as beta-amyloid and tau proteins, compounds linked to Alzheimer's disease. This network, known as the glymphatic system, operates most effectively during slow-wave sleep. Even modest sleep loss impairs its performance. Regular, unbroken deep sleep is therefore one of the simplest and most powerful forms of neuroprotection known to science. Metabolism follows the same nightly rhythm. Insulin sensitivity increases during the day and decreases after dark, meaning late-night eating forces the pancreas to work against its natural schedule. During sleep, cells switch from energy consumption to repair, oxidizing fat and recycling proteins. Chronic sleep deprivation disrupts this cycle, raising cortisol and inflammatory markers while lowering leptin, the hormone that signals fullness. Over time, the imbalance promotes weight gain and accelerates aging. Restoring proper rhythm reverses these effects. Balanced hormones, stable glucose, calmer immunity. Emotionally, sleep reorganizes the mind. The limbic system, where feelings are processed, connects with the prefrontal cortex, allowing emotional experiences to be integrated without overwhelming awareness. Dreams weave fragments of memory into narratives that reduce anxiety and strengthen adaptation. A well-rested brain approaches each day with lower baseline stress, preserving cognitive and cardiovascular stability. Mental calm, achieved through regular sleep, is therefore a biological asset, not a luxury. Different tissues display their own circadian patterns of restoration. The liver processes toxins, the gut recalibrates microbial composition, and the immune system releases cytokines that coordinate repair. During this time, inflammatory signals decline, oxidative defenses rise, and cellular debris clears. Even skeletal muscle uses sleep to rebuild from microscopic damage. Without these nightly renewals, small inefficiencies multiply into chronic disease. The connection between sleep and longevity is now unmistakable. People who consistently sleep 7 to 8 hours per night show lower mortality and reduced risk of dementia, heart disease, and metabolic disorders. Short sleepers, by contrast, accumulate physiological debt that no amount of caffeine or motivation can repay. The equation is simple. The more consistent the restoration, the slower the wear. If you are listening while resting now, let your body settle into its own natural rhythm. Breathing deepens, muscles loosen, and the mind drifts toward quieter thought. That gentle descent is not wasted time. It is repair in motion, life renewing itself in silence. Every moment of genuine rest restores the balance between energy and calm, allowing cells to remember what youth once meant. When you sleep tonight, remember, this is longevity's most faithful ally. The body heals not through effort but through surrender to its rhythm, night after night, breath after breath. The rhythm of emotion marks biological time as clearly as any clock. Every surge of joy, fear, or sorrow changes pulse, breath, and hormone flow, nudging the body slightly forward or backward in its aging curve. Where chronic tension compresses time, calm perception stretches it. In this sense, longevity is partly psychological, the product of how we experience and regulate emotion across years. The body's first responder to emotion is the autonomic nervous system. When the brain detects threat or challenge, the sympathetic branch releases adrenaline and noradrenaline, quickening heart rate and preparing muscles for action. If the threat fades, the parasympathetic branch restores equilibrium, lowering blood pressure and calming the heart. Problems arise when the first signal never truly stops. Persistent worry, competition, or exposure to constant noise keeps the accelerator pressed, flooding tissues with cortisol and inflammatory cytokines. This low-grade arousal erodes repair, suppresses immunity, and accelerates vascular aging. Stress becomes a slow-burning chemical weathering of the body. Short bursts of stress, however, are not harmful. they are instructive. Controlled exposure, exercise, creative challenge, even public speaking, stimulates hormetic responses that strengthen resilience. What matters is recovery. Each return to calm rehearses the body's ability to end its own alarm. Longevity, then, is not the absence of stress, but the mastery of recovery speed. The quicker the return to baseline, the less cumulative wear emotional storms leave behind. Neurochemistry links feeling to physiology. Serotonin shapes mood stability, dopamine regulates motivation and reward, and oxytocin supports trust and bonding. Balanced levels of these messengers maintain social connection and optimism, traits repeatedly correlated with extended lifespan. Isolation or chronic conflict, conversely, alters these circuits, lowering oxytocin and raising cortisol. Over years, that imbalance fosters inflammation and metabolic strain. Emotional health, though invisible, translates directly into molecular order. The brain structures governing emotion reveal how perception shapes biology. The amygdala scans for threat, the prefrontal cortex interprets and moderates its reactions. Mindfulness and cognitive reframing strengthen the cortical pathways that restrain panic, teaching the brain to pause before reacting. With practice, the amygdala quiets faster, cortisol pulses shorten, and heart rate variability, a key marker of resilience, improves. Each moment of awareness rewires stress circuits, buying seconds of calm that accumulate into years of health. Cultural and social context amplify or buffer emotional load. Communities that emphasize cooperation and belonging show lower stress hormones and slower cognitive decline. Shared rituals, meals, music, conversation act as collective regulators, synchronizing breathing and heartbeat among participants. This social coherence lowers physiological arousal in ways comparable to meditation. Longevity flourishes where human connection remains strong, reminding biology that safety still exists. Even perception of time itself is emotional. Under pressure, time seems to rush. In calm focus, it expands. This subjective elasticity mirrors biochemical states. High adrenaline compresses experience into urgency, while parasympathetic dominance lengthens perception, allowing deeper restoration. People who cultivate mindful presence through art, nature, or contemplation often report that time feels slower and richer. Their physiological markers agree: lower resting heart rate, improved immune balance, steadier blood glucose. The sense of having more time may, at a cellular level, correspond to actually using time more efficiently. Chronic negative emotion, left unresolved, expresses itself in posture, muscle tone, and breathing. Shoulders tighten, diaphragm shortens, oxygenation drops. Over decades, these physical adaptations harden into patterns that strain joints and restrict circulation. Emotional release, whether through therapy, breathing, or music, literally reopens the body's mechanical rhythm, improving flow and nutrient delivery. The mind and body share one timeline. Harmony in one slows the wear of the other. In biological terms, emotion is energy distribution. Positive affect spreads energy evenly, supporting maintenance. Persistent fear localizes it in muscles and glands, draining reserves meant for repair. Longevity arises when emotion becomes information rather than residue, felt fully, then released. The nervous system resets, hormones return to baseline, and the clock continues at its natural pace. When calm becomes the default state, stress no longer defines identity but serves as momentary alertness. The years stretch quietly forward, marked not by urgency but by equilibrium. In that composure, biological time itself seems to breathe more slowly. Every heartbeat, thought, and movement draws on energy generated deep inside the cell. That energy originates in tiny organelles called mitochondria, descendants of ancient bacteria that once lived freely but were absorbed by early cells billions of years ago. Through this symbiosis, they became the engines of complex life, turning food and oxygen into usable fuel. Longevity depends on how well these engines perform and how smoothly they coordinate their output with the body's demands. Mitochondria produce adenosine triphosphate, ATP, through a process known as oxidative phosphorylation. Within their folded inner membranes, Electrons flow along a chain of proteins, releasing energy that drives the synthesis of ATP. A fraction of those electrons, however, escape and react with oxygen to form reactive oxygen species, unstable molecules that damage proteins, lipids, and DNA. In youth, antioxidant enzymes such as superoxide dismutase and catalase neutralize most of these radicals. With age, efficiency declines, and oxidative noise increases. The art of living long is the art of keeping this energy conversion both powerful and clean. Cells regulate mitochondrial activity with remarkable precision. When energy demand rises, during movement, thought, or stress, mitochondria multiply, fusing into larger networks that distribute ATP rapidly. When demand falls, they divide and rest. This constant remodeling, called mitochondrial dynamics, prevents stagnation and ensures that damaged components are isolated and recycled through mitophagy. Exercise, fasting, and thermal variation all stimulate this process, effectively renewing the cell's power plants. Sedentary life, in contrast, lets broken mitochondria linger, leaking free radicals and triggering inflammation. Genetics and environment jointly shape mitochondrial resilience. Mutations in mitochondrial DNA accumulate over time, since each organelle carries its own small genome. These mutations impair energy output and signal distress to the rest of the cell. Antioxidant nutrients, coenzyme Q10, alpha-lipoic acid, and polyphenols from plants help buffer these effects, supporting electron flow and preserving membrane integrity. Yet the most powerful stimulus for mitochondrial renewal remains metabolic challenge, alternating periods of activity and rest, abundance and scarcity. The mitochondrion thrives on variation. Constancy dulls its responsiveness. Energy metabolism also links directly to hormonal balance. Thyroid hormones regulate how quickly mitochondria burn fuel. Insulin dictates how readily glucose enters cells. Cortisol and adrenaline fine-tune distribution during stress. When these messengers lose rhythm, energy becomes chaotic. Too much in some tissues, too little in others. Chronic overnutrition, for example, floods mitochondria with excess substrates, overwhelming their capacity and generating oxidative waste. Moderate intake, regular movement, and steady circadian timing restore balance, ensuring that mitochondria operate in synchrony with hormonal cues. Within the brain, mitochondrial health dictates clarity of thought. Neurons consume immense energy yet cannot tolerate oxidative overload. Mitochondria in neural tissue coordinate with calcium channels to regulate firing patterns and memory formation. Sleep, as established earlier, allows these organelles to clear waste and reset metabolism. Even small deficits in mitochondrial efficiency can slow cognition or mood, showing how intimately the mind's endurance relies on cellular engines. Beyond energy production, mitochondria act as signaling hubs. They influence apoptosis, programmed cell death, immune responses, and even stem cell behavior. When energy falls below critical levels, they release molecules that alert the nucleus to adjust gene expression, a conversation known as retrograde signaling. This feedback loop integrates cellular status with global physiology. In essence, mitochondria translate the language of metabolism into the language of lifespan. Recent studies suggest that mild mitochondrial stress may extend longevity by activating defense pathways such as the mitochondrial unfolded protein response. When slightly challenged, mitochondria send signals that increase repair and antioxidant production throughout the cell. This adaptive response mirrors hormesis at the microscopic level. Brief discomfort yields long-term strength. The key, again, is proportion. Stress measured in minutes, not months. Metabolism as a whole reflects the harmony between fuel intake and energy demand. When these forces align, ATP flows easily, heat dissipates smoothly, and cells remain flexible. When imbalance persists, through constant eating, inactivity, or unrelieved tension, the system clogs with byproducts, and entropy accelerates. Longevity arises from rhythm, periods of effort and replenishment, feeding and fasting, motion and rest. Within that rhythm, mitochondria stay alert and young. The quiet miracle of these organelles is that they still carry traces of their ancient independence. They remind every cell that cooperation sustains power longer than domination. When mitochondria function in harmony, energy moves without friction, and life proceeds with calm precision. The measure of age, at the deepest level, is not years counted, but how efficiently these tiny engines continue to hum. For all its ingenuity, life remains bound by physical law. Molecules break, entropy rises, and time reshapes even the most carefully maintained structure. Yet throughout history, humans have refused to accept these boundaries as absolute. From the first myths of elixirs and fountains to the laboratories of modern biogerontology, the question endures: is there an upper limit to how long a body can last? Current data suggest that the human lifespan reaches a natural ceiling near 120 years. Beyond that threshold, survival curves flatten, not because of a single failure, but because multiple systems begin to lose coordination at once. DNA repair slows, stem cell reserves thin, immune precision fades, and energy balance falters. No single mechanism breaks. Rather, the network unravels. This interdependence defines the complexity of aging. One component cannot remain young while others decline. Longevity science therefore seeks not eternal youth but synchronized preservation, keeping the entire orchestra in tune for as long as possible. Researchers explore several strategies to extend this harmony. Genetic modulation of key pathways, mTOR, AMPK, sirtuins, and insulin signaling has already lengthened lifespan in animals. Senolytic therapies clear away senescent cells that pollute tissues with inflammatory signals, while stem cell rejuvenation replenishes the body's repair crews. Advances in epigenetic reprogramming promise to reset molecular clocks, restoring youthful gene expression without erasing identity. Meanwhile, AI-driven bioinformatics analyze immense datasets to uncover patterns linking environment, behavior, and longevity at scales no human intuition could track. Each approach targets a different layer of the same architecture: damage, regulation, and coordination. Still, extending life raises profound questions about purpose and balance. Biology evolved under the assumption of turnover, generation replacing generation to preserve diversity. If lifespan stretches far beyond current limits, societies must adapt not only medically but psychologically. What meaning does growth hold when decades stretch into centuries? How do memory and motivation function when time itself dilates? Science can slow decline, but philosophy must decide what kind of existence extended years should contain. The frontier of longevity research increasingly merges biology with engineering. Organs grown from patient-derived cells, nanobots designed to clear molecular waste, and smart implants that monitor metabolic signals hint at bodies that repair themselves automatically. Some envision integration of digital systems with biological memory, allowing minds to persist beyond fragile neurons. Others pursue cryopreservation or mind uploading, concepts still speculative but emblematic of humanity's refusal to let mortality stand unchallenged. Whether these pursuits will deliver genuine continuity or only simulation remains open. What becomes clear is that the pace of discovery accelerates. The time between the identification of a molecular pathway and the creation of a targeted therapy shortens with each decade. This compression of scientific time mirrors the compression of biological time we seek to resist. Yet, even as the tools sharpen, the fundamentals persist: balanced energy, restorative sleep, emotional calm, clean environment, and connection. Every advanced intervention ultimately attempts to mimic what nature already perfected: coherence among systems. There are likely practical ceilings rooted in thermodynamics and information theory. Cells cannot maintain perfect fidelity indefinitely. Random fluctuations guarantee eventual error, even if damage repair approaches perfection. Stochastic noise ensures entropy's gradual return. The aim, therefore, is not immortality but asymptotic youth, approaching stability ever more closely without reaching infinity. The limit is not a wall but a horizon, receding as understanding deepens. In contemplating that horizon, a quiet humility arises. Each additional year of health is not conquest, but dialogue with the forces that created us. The body remains an experiment in equilibrium, negotiating constantly with gravity, radiation, and chemistry. Extending life becomes meaningful only when those added years expand awareness, compassion, and contribution rather than mere duration. Perhaps longevity's truest future lies not in defying nature but in aligning with it more consciously, living in a way that minimizes friction with the world's rhythms while maximizing understanding of its design. When science and wisdom converge on that point, the upper limit of life may no longer feel like an end but a seamless continuation of balance. Longevity is not a contest against time. It is a dialogue between persistence and change, between the drive to endure and the wisdom to adapt. Every organism, from the simplest bacterium to the most complex human being, participates in this conversation. Life's success has never been measured only by duration but by coherence, how smoothly energy, structure, and meaning remain aligned through the years. At the molecular scale, we see constant renewal. DNA repaired, proteins recycled, mitochondria replaced. At the cellular scale, growth yields to maintenance, and maintenance to graceful decline. Yet decline itself is not failure. It is nature's way of transferring knowledge from one generation of cells to the next. Each heartbeat carries a rhythm inherited from billions of ancestors who perfected endurance through impermanence. Longevity is their legacy written into our chemistry. Viewed through this lens, time becomes less an enemy than a medium for refinement. The longer an organism lives, the more precisely it learns to manage balance between stress and calm, between abundance and scarcity, between isolation and connection. The aim is not to freeze youth but to extend clarity, a state in which every cell, tissue, and thought cooperates with minimal waste and maximal understanding. In that equilibrium, life feels neither hurried nor delayed. It simply continues. Human beings, unlike any other species, can reflect on this process. We study our biology, alter our environment, and imagine futures beyond current limits. Yet each discovery reminds us of the same truth that nature repeats. Endurance depends on rhythm. The alternation of activity and rest, of waking and sleeping, of curiosity and contemplation, these are not interruptions to living but its essential structure. As science edges closer to reversing aging at the molecular level, a deeper insight emerges. The quest for longevity ultimately mirrors the quest for balance within. The hormones, neurons, and genetic codes that sustain life also reflect our emotional and philosophical state. A mind at peace generates chemistry of repair. A body in harmony fosters clarity of thought. Health and awareness are not separate goals but two sides of the same stability. The future of longevity will likely blend technology with timeless wisdom, engineered tissues, targeted therapies, and perhaps even selective reprogramming of cells to renew the body from within. But these advances will matter only if they preserve the underlying rhythm, the alternation of renewal and rest that has guided life since its origin. Without that rhythm, even endless existence would feel unfinished, a machine running without music. Tonight, as you rest, remember that every breath you take participates in this ancient cycle. The slowing of your heart, the deepening of your breath, and the quiet unfolding of sleep are not signs of weakness but acts of profound intelligence. In stillness, the body listens to itself and restores its design. The hours of night do what no medicine can. They return order to the system, teaching each cell to remember what harmony feels like. In the calm between one heartbeat and the next, longevity ceases to be a pursuit and becomes a presence, the silent confidence that life, when balanced, knows how to continue. We from Sleepy Stories wish you a good night.